कि जब वो इस हॉल में प्रवेश करें तो हम सभी उनको एक स्टैंडिंग ओवेशन दें। सभी से अनुरोध है कि वो अपना स्थान ग्रहण करें। आज के मुख्य अतिथि परम आदरणीय पद्म भूषण डॉक्टर R.S. परोदा सर, चेयरमैन टास, फॉर्मर डायरेक्टर। जी सर, थोड़ा वॉल्यूम बढ़ाइए प्लीज। हेलो हेलो, थोड़ा वॉल्यूम बढ़ाइए। हाँ बस। आज के मुख्य अतिथि परम आदरणीय पद्म भूषण डॉक्टर R.S. परोदा सर, चेयरमैन टास, फॉर्मर डायरेक्टर ICAR-NBPGR, फॉर्मर सेक्रेटरी डेयर एंड DG ICR। आदरणीय विशिष्ट अतिथि एवं चेयरमैन डॉक्टर ML जाट सर, सेक्रेटरी डेयर एंड DG ICR। आदरणीय विशिष्ट अतिथि श्री ज्ञानेंद्र त्रिपाठी जी, एडिशनल सेक्रेटरी डेयर एंड सेक्रेटरी ICR। माननीय विशिष्ट अतिथि एवं आमंत्रित वक्ता डॉक्टर नागेंद्र कुमार सिंह जी, JC बोस नेशनल फेलो, फॉर्मर नेशनल प्रोफेसर एंड BP पाल चेयर ICR NIPB। माननीय डॉक्टर DK यादव जी, निदेशक ICAR-NBPGR एंड DDG क्रॉप साइंस। मंचासीन आदरणीय डॉक्टर RK गौतम जी, अध्यक्ष जर्मप्लाज्म मूल्यांकन प्रभाग NBPGR। अन्य गणमान्य अतिथिगण, सम्मानित वैज्ञानिकगण, कर्मचारी, छात्र, उपस्थित सहकर्मीगण, हमारे क्षेत्रीय केंद्रों से ऑनलाइन जुड़े सभी अधिकारी एवं कर्मचारीगण तथा उपस्थित सभी महानुभाव, आप सबको सादर नमस्कार। ICAR राष्ट्रीय पादप आनुवंशिक संसाधन ब्यूरो के 51वें स्थापना दिवस समारोह में आप सभी का हार्दिक स्वागत एवं अभिनंदन है। कार्यक्रम का शुभारंभ राष्ट्रभक्ति एवं मातृभूमि के प्रति सम्मान की भावना के साथ वंदे मातरम से किया जाएगा। मैं सभी उपस्थित जनों से विनम्र अनुरोध करती हूँ कि कृपया अपने स्थान पर खड़े हो जाएं और राष्ट्रगीत में सहभागी बने। निदेशक एनबीपीजीआर डॉक्टर डीके यादव जी से अनुरोध करती हूँ कि वे पौधे भेंट कर हमारे माननीय अतिथियों का स्वागत करें। धन्यवाद सर। अब मैं आदरणीय मुख्य अतिथि, विशिष्ट अतिथियों एवं मंचासीन सभी गणमान्य जनों से सादर अनुरोध करती हूँ कि वे दीप प्रज्वलन कर इस समारोह का विधिवत शुभारंभ करें एवं डॉक्टर एचबी सिंह जी के चित्र पर पुष्पांजलि अर्पित करें। तालियों का सिलसिला रुकना नहीं चाहिए। धन्यवाद। संसाधन ब्यूरो के 51वें स्थापना दिवस का उत्सव मना रहे हैं, जो पादप आनुवंशिक संसाधनों के संरक्षण, प्रबंधन, आदान प्रदान और सतत उपयोग के क्षेत्र में 5 दशकों से भी अधिक समय से कार्य कर रहा है। आज का यह अवसर हम सभी के लिए अत्यंत गौरव, हर्ष और आत्ममंथन का क्षण है। किसी भी संस्थान का स्थापना दिवस केवल उसकी स्थापना की वर्षगांठ ही नहीं होता, बल्कि यह उसके गौरवशाली इतिहास, उपलब्धियों, मूल्यों तथा भावी संकल्पों का उत्सव भी होता है। यह अवसर हमें उन सभी व्यक्तियों के प्रति कृतज्ञता व्यक्त करने का अवसर प्रदान करता है, जिनके समर्पण, परिश्रम और दूरदृष्टि ने संस्थान को निरंतर प्रगति और उत्कृष्टता के पथ पर अग्रसर किया है। इन्हीं भावनाओं के साथ मैं माननीय निदेशक डॉक्टर डीके यादव जी से विनम्र अनुरोध करती हूँ कि वे हमारे माननीय मुख्य अतिथियों का एवं अन्य गणमान्य अतिथियों का स्वागत करें तथा वर्ष के दौरान ICAR-NBPGR की प्रमुख उपलब्धियों एवं उल्लेखनीय प्रगति से हम सभी को अवगत कराएं।
थैंक यू वर्तिका। सभी को नमस्कार। और आज के इस 91वें फाउंडेशन डे पर मैं आप सभी को हृदय से बधाई देता हूं। और आज जो हमारे संस्थान के 50 वर्ष कल पूरे हुए हैं, तो उस पर भी आप सभी को विशेष तौर पे बधाई देता हूँ। और मैं सभी हमारे वर्तमान स्टाफ, हमारे निदेशकगण, आईसीआर के सीनियर्स जो पूर्व में इस ब्यूरो के साथ शुरू से रहे, खास तौर पे डॉक्टर आर.एस. परोदा साहब का लंबा एक संस्थान के साथ जुड़ाव रहा, मैं उन सभी को बहुत बहुत धन्यवाद देता हूं कि आप इस संस्थान को एक विश्व पटल पर लाने में बहुत ही आप लोगों की मदद रही। और आज हम जिस स्थिति में हैं, तो हमारे सभी जितने भी पूर्व में अधिकारी कर्मचारी रहे, उन सभी का बहुत बहुत योगदान रहा है। आज के इस कार्यक्रम के मुख्य अतिथि पद्म भूषण प्रोफेसर आर.एस. परोदा साहब, former Secretary Dear and Director General ICR, as well as currently the Chairman of Trust for Advancement of Agriculture Sciences, our respected Secretary Dear and Director General Dr. M.L. Jad Saab, our guest of honor, our very dynamic Secretary ICR and Additional Secretary Dear श्री ज्ञानेंद्र देव त्रिपाठी जी, हमारे सीनियर कुलीग और आज के डॉक्टर हरभजन सिंह के जो लेक्चर के लिए आमंत्रित किए हैं, हमारे बहुत ही प्रखर वैज्ञानिक डॉक्टर एन.के. सिंह साहब, मेरे साथी डॉक्टर गौतम साहब, हमारे फॉर्मर डायरेक्टर्स में डॉक्टर एस.के. शर्मा साहब ऑनलाइन जुड़े हुए हैं। उसके अलावा जितने भी हमारे जो पूर्व में साथी रहे, मैं देखता हूँ बड़ी संख्या में अभी कुछ कार्यरत हैं, कुछ हो गए। डॉक्टर अग्रवाल अनुराधा हैं, डॉक्टर वीना, उधर से नाम लेता हूँ, डॉक्टर भागमल जी, डॉक्टर श्रीवास्तव साहब, डॉक्टर करियालू जी, डॉक्टर अशोक कुमार, डॉक्टर राणा जी, डॉक्टर मलिक साहब, डॉक्टर बी.एम. सिंह साहब, डॉक्टर सुधीर कोचर, डॉक्टर अलावत, डॉक्टर मलिक पीछे बैठे हैं, और कुछ मेरे को लगता है मेरे नज़र नहीं आ रहे, तो जो नज़र आ रहे हैं, तो मैं आप सभी का हृदय से स्वागत करता हूं। और आज आप इस कार्यक्रम में सभी शामिल हुए, उसके लिए मैं आप सभी का बहुत धन्यवाद करता हूं। डॉक्टर रोहिणी नायर हैं ऑनलाइन, डॉक्टर प्रतिभा और डॉक्टर गुरिंदर जुड़े हुए हैं, तो मैं आप सभी का हृदय से स्वागत करता हूं। इसके साथ ही हमारे संस्थानों के ज्वाइंट डायरेक्टर डॉक्टर विश्वनाथन हैं, प्रोजेक्ट डायरेक्टर डॉक्टर ब्रह्मानंद जी, सही हमारे साथी डॉक्टर सुनील, राजश्री सुनील और उनकी टीम, तथा संस्थान के सभी वर्तमान में और पूर्व के अध्यक्ष, पांचों डिविजन्स के हमारे हेड, जो यूनिट्स के इंचार्जेज, सभी हमारे एनबीपीजीआर परिवार और आईसीआर परिवार के सभी साथियों, वैज्ञानिकगण, फाइनेंस, टेक्निकल, एडमिनिस्ट्रेशन, स्टूडेंट्स, जितना भी हमारे स्टूडेंट्स आ रहे हैं सारे, तथा जितना भी कॉन्ट्रेक्चुअल स्टाफ है, आप सभी का मैं हृदय से स्वागत करता हूं और आप सभी को बहुत बहुत इस शुभ अवसर पर बधाई देता हूं। आप सभी जानते हैं कि हमारे देश में जो पीजीआर की एक्टिविटीज 1910 से शुरू हुई, तो उसके बाद जो सिस्टमेटिक 1946 में जो हमारा सेक्शन जब बना बोटनी डिवीजन, उसमें जो इंपीरियल एग्रीकल्चर रिसर्च इंस्टीट्यूट में 1961 में जो डिवीजन ऑफ प्लांट इंट्रोडक्शन IRI में एस्टेब्लिश हुआ, जिसको लेट हरभजन सिंह जी ने लीड किया, एंड टू द जर्म प्लान एक्सप्लोरेशन, कलेक्शन एंड मेंटेनेंस. In 1976, this plant introduction division was elevated to the current National Bureau of Plant Genetic Resources. You all are well aware, and it has a very strong network with 10 regional stations and 5 divisions and 4 units here at the headquarters. The National Gene Bank was established in 1996, and you all know that today, with the establishment of this gene bank, we are the second largest globally, having the around 4.75 lakh accessions in our national gene bank. सॉरी फॉर द डिस्टर्बेंस। और जितना कंजर्वेशन की हमारी एक्टिविटीज आप सभी जानते हैं, चार तरह से हम कंजर्वेशन की एक्टिविटी चला देते हैं। सीड जीन बैंक जो कि -18 डिग्री सेंटीग्रेड पे, क्रायो जीन बैंक -170 टू -196, इन विट्रो जीन बैंक 25 डिग्री सेंटीग्रेड पे है, और जो फील्ड जीन बैंक है हमारे, तो जो लॉन्ग टर्म और मीडियम टर्म स्टोरेज का जितना भी देश के जेनेटिक रिसोर्सेज हैं, प्लांट जेनेटिक रिसोर्सेज पे पूरा कार्य करते हैं। 1984 में सिर्फ 285 जो सीड सैंपल्स के साथ हमने जीन बैंक को शुरू किया, और आज जैसे कि मैंने बताया कि विश्व के दूसरे नंबर का जीन बैंक जिसमें 4.7 लाख एक्सेशंस हैं, और जिसमें से लगभग 1 लाख जो है, हम बात ट्रेडिशनल वैरायटीज की, लैंड रेसिस की बात करते हैं, पोल्क वैरायटीज की बात करते हैं, तो वो भी इन 4.75 लाख में उपस्थित है। और ये एक बार बार इस तरह के सवाल उठते हैं कि हमारी जो पुरानी डाइवर्सिटी थी, जेनेटिक डाइवर्सिटी थी, वो लुप्त होती जा रही है, कोई ध्यान नहीं दिया जा रहा। तो लगभग 25%, 20-22% के आसपास जितना भी हमारा जेनेटिक रिसोर्स है, उसमें हमारा जो ट्रेडिशनल वैरायटी, लैंड रिसोर्स वैरायटीज, वो सभी हमने कंजर्व की हुई है। और आप सभी जानते हैं कि हमारे जीन बैंक की कैपेसिटी 10 लाख एक्सेशंस की है, जिसमें अभी हम 50% पर भी नहीं पहुंचे हैं। इसके साथ ही जो हमने 59 जो है हमारे विभिन्न संस्थानों में, विभिन्न कृषि विश्वविद्यालयों में एक नक्सल एक्टिव जर्म प्लांट साइट्स है, इनको हमने नोटिफाई किया है। और पिछले 4-5 साल पहले इनको कोई फाइनेंशियल सपोर्ट हम नहीं दे पाते थे, तो बार बार संस्थानों की एक शिकायत रहती थी कि जितना भी जनरली जो हॉर्टिकल्चर क्रॉप्स के जो जर्म प्लांट को मेंटेन करते हैं, तो उसकी मेंटेनेंस की एक बड़ी समस्या थी, और उस समस्या को ओवरकम करने के लिए इसको शुरू किया लगभग 3 साल पहले, और आज हम सभी को सपोर्ट करते हैं और उसको अच्छी तरह से मेंटेन करते हैं। एक प्लांट जर्म प्लांट रजिस्ट्रेशन कमेटी के द्वारा ये भी हमारे माननीय डॉक्टर प्रोदर साहब की एक पहल पे ये कार्य शुरू किया गया, और अभी तक हमने उसमें लगभग 2432 जो ट्रेड स्पेसिफिक जर्म प्लाज्म की बात करते हैं, तो उनको रजिस्टर किया, और उनमें लगभग 240 स्पीशीज, डिफरेंट स्पीशीज की ये जर्म प्लाज्म है, जिनको कि हम क्रॉप इम्प्रूवमेंट में बड़ा फ्रीक्वेंटली शेयर करते हैं। और जो कोई भी साइंटिस्ट नए सिरे से शुरू करता है, तो उसको जीरो से स्टार्ट करने की बजाय एक रेडीमेड मटेरियल क्रॉप वाइज अवेलेबल होता है, जिसको अपने ब्रीडिंग प्रोग्राम में यूज कर सकते हैं। हमारा जो ब्यूरो है वो जितना भी जर्म प्लाज्म इम्पोर्ट एक्सपोर्ट की एक नोडल एजेंसी है, और जितने भी इम्पोर्ट एक्सपोर्ट होते हैं, तो हमारी जर्म प्लाज्म पॉलिसीज एंड एक्सचेंज यूनिट और क्वारंटाइन डिवीजन, इनका एक अहम रोल रहता है। और सारी पब्लिक प्राइवेट सेक्टर, जितने भी प्राइवेट सेक्टर है या हमारे पब्लिक एजेंसीज हैं, स्टेट एग्रीकल्चर यूनिवर्सिटीज हैं, उनको हम फैसिलिटेट करते हैं। ब्यूरो का जो नेशनल लेजिस्लेशन में एक बहुत ही बड़ा रोल रहा है, चाहे आप ICAR-NBPGR की बात करें, चाहे हम बायोडाइवर्सिटी एक्ट की बात करें, चाहे सीड बिल की बात करें, तो जितने भी रेगुलेशंस हैं प्लांट जर्म प्लाज्म एक्सचेंज के, प्लांट जर्म प्लाज्म कंजर्वेशन के, तो उनमें ब्यूरो का एक बहुत ही महत्वपूर्ण योगदान रहता है, और उसके लिए ब्यूरो की एक अपनी पहचान है। यहाँ तक पिछले वर्ष की कुछ गतिविधियों की बात है, तो हमने पिछले साल लगभग 16 एक्सप्लोरेशन किए एक्सप्लोरेशन डिवीजन ने, जिसमें 1261 डिफरेंट जो एक्सेशंस हम कलेक्ट कर पाए, जिसमें 788 एक्सेशंस वो कल्टिवेटेड स्पीशीज के थे, और 433 वाइल्ड स्पीशीज थे। और ये एक ही बहुत ही एग्री हॉर्टिकल्चर क्रॉप्स का बड़ा यूनिक जर्म प्लांट, जिसमें से एक तो नया टेक्सा जो आइडेंटिफाई किया, जो एजिप्टिका वैरायटी है, जो कि एक नया एडिशन है। और उसके अलावा इसमें हमने सिस्टमेटिक्स का भी कार्य शुरू किया, जो कि इससे पहले उस पर बहुत अधिक ध्यान नहीं दिया जा रहा था। एक जो नई पहल जो की गई, उसमें जो 20 कम्युनिटी सीड बैंक स्थापित किए गए, जिसमें कि हम काफी समय से हम संघर्ष कर रहे थे, तो हमारे जो एक्सप्लोरेशन डिवीजन ने एक नई पहल करके 20 कम्युनिटी सीड बैंक की भी स्थापना की। जर्म प्लाज्म एक्सचेंज की बात करें, तो क्योंकि ब्यूरो का एक मुख्य स्तंभ ये भी है, जिसमें हमने 61,515 एक्सेशंस को इंपोर्ट किया, 7 एक्सपोर्ट्स को फैसिलिटेट किया। उसके अलावा 77,904 जो सैंपल वो हमने विभिन्न रिसर्चर्स को जो मटेरियल ट्रांसफर एग्रीमेंट के लिए कृषि विश्वविद्यालयों में, हमारे संस्थानों में, और जितने भी प्राइवेट सेक्टर के कोलीग्स थे, उनसे शेयर किया। और 464 टोटल इम्पोर्ट परमिट जो है वो हमने इशू किए। गर्मप्लान क्वारंटाइन का जहां तक सवाल है, तो ये पूरे देश की सुरक्षा, फूड सुरक्षा का एक तरह से मूल हमारा डिवीजन है कि जितना भी इम्पोर्ट हमारा होता है, तो उसके लिए जब तक क्वारंटाइन की गतिविधियां सही नहीं होगी, तो निश्चित रूप से ये हमें पूरे देश के लिए बड़ा खतरा हो सकता है। तो टोटल 1,21,887 सैंपल्स जो हमने विभिन्न स्रोतों से इम्पोर्ट किए, तो इनका पूरा जितना भी पोस्ट रिलीज का जो क्वारंटाइन की एक्टिविटीज क्वारंटाइन डिवीजन ने की, और टोटल 47 फाइट और सैनिटरी सर्टिफिकेट्स लगभग 7,035 सैंपल्स के लिए जो एक्सपोर्ट में प्राइवेट कंपनी ज्यादातर उसको डील करती है, तो उसके लिए कार्य किया। और टोटल इन एक्टिविटीज से जो रेवेन्यू जनरेशन 3.15 करोड़ का हुआ, और टोटल रेवेन्यू जनरेशन बाकी एक्टिविटीज इन फिंगर प्रिंटिंग वगैरह लगा के पिछले साल का लगभग 4 करोड़ का रेवेन्यू जनरेशन ब्यूरो का रहा। गर्म प्लान करेक्टराइजेशन इवेल्युएशन एक बहुत ही आवश्यक गतिविधि है, और पीछे अभी हमारा जो नेशनल एडवाइजरी बोर्ड फॉर मैनेजमेंट ऑफ प्लांट जेनेटिक रिसोर्सेज की एक मीटिंग 29 तारीख को हुई थी। माननीय डॉक्टर परोदा साहब उसके अध्यक्ष हैं, और क्योंकि ये लगभग 10 वर्षों से ये जो एडवाइजरी बोर्ड था ये डिफंक्ट था, और माननीय परोदा साहब के इनिशिएटिव पे और इनके डायरेक्शन पे हमने इसको रिवाइव किया। तो उसमें जो हमारे 6 ब्यूरो हैं, उनके सभी जो निदेशक उपस्थित थे, और ब्यूरो में लगभग इस बोर्ड में लगभग 31 विभिन्न विभागों से वैज्ञानिक और जो जितने भी मैनेजर्स इसमें इन्वॉल्व हैं। तो इसमें एक बात ये जरूर एक मुद्दा रहा कि हम कितने जन्मकाल को 4.75 की जब हम बात करते हैं लाख की, तो उसमें से कितने हम प्रैक्टिकली रिसर्च में यूज कर रहे हैं, करेक्टराइजेशन कर रहे हैं। तो इसमें निश्चित रूप से सर पिछले 4-5 सालों में एक बहुत अच्छी हमने प्रगति की। और लास्ट ईयर की जो बात करते हैं, तो 27,948 एक्सेशन विभिन्न क्रॉप्स की हमने इनको करेक्टराइज किया, फील्ड वैल्यूएशन किया और रीजनरेट किया, जिसको कि हम दोबारा जीन बैंक में जो है ले जाने का कार्य अभी कर रहे हैं। गर्म प्याज कंजर्वेशन का जैसे कि मैंने बताया कि पिछले साल जो है 2,074 टोटल एक्सेशन आए, और ये मीटिंग के विषय का चर्चा हुई कि जो हमारा जो इलेक्शन की जो गति है वो भी धीमी हुई है, और जो वैज्ञानिक जो जितना गर्म प्याज साइंटिस्ट पिछले 5 वर्षों में उस पर मेरे विचार से हमें कार्य करने की आवश्यकता है। और मैंने माननीय परोदा साहब को मैंने प्रोमिस किया है कि हम इस गतिविधि को जितने भी हमारे संस्थानों के थ्रू और बाकी उसमें हम उसको आगे बढ़ाएंगे। जर्म प्लान रजिस्ट्रेशन, जैसे कि मैंने कहा कि जो ट्रेड स्पेसिफिक जर्म प्लान की हम बात करते हैं, तो एक अच्छी एक्टिविटी है। लास्ट ईयर हमने 111 जो 2,400 जो मैं 32 की बात कर रहा हूँ, तो उसमें 111 नए जर्म प्लान हमने जो डोनर जो आइडेंटिफाई किए और उनको इस एक्टिविटी में हमने शामिल किया। डीएनए फिंगर प्रिंटिंग का, क्योंकि पहले उस दिन भी चर्चा हुई कि पहले हमारा डीएनए फिंगर अलग से एक सीआरपी थे, डॉक्टर गढ़वाल साहब वहाँ बैठे हैं, लेकिन धीरे धीरे उसकी गतिविधियां कम हुई और वो अभी एक जीनोमिक रिसोर्सेज डिवीजन के रूप में कार्य करते हैं। और इसका एक मुख्य कार्य जो जीनोमिक रिसोर्सेज एक्टिविटीज के अलावा जितनी भी वैरायटीज आइडेंटिफाई होती है सीडेक 96 के सेक्शन 5 के अनुसार, तो उन सभी का डीएनए प्रोफाइलिंग एक डेटा की आवश्यकता पड़ती है। तो ये कार्य ये हमारा डिवीजन करता है। और जीएमओ टेस्टिंग के लिए 2 ही नोटिफाइड लैब है आईसीआर में, एक आईसीआर कानपुर में, एक हमारी लैब है जो कि एनएबीएल एक्रेडिट है। और इस पर भी सारा काम लगभग 53 सैंपल विद एजेंसी से आए, उसका हमने जो है विश्लेषण किया। एक पेटेंट भी यहाँ से ग्रांट किया गया है, जिसमें एक एक चिप जीन चिप 64 के एसएनपी चिप ग्रेन एम्ब्रेंट के लिए हमने डेवलप की है, और उसकी लाइसेंसिंग की प्रक्रिया चल रही है। एआईएनपी ऑन पोटेंशियल क्रॉप, जैसे बार बार हमारे डीजी साहब भी और परसों की मीटिंग में भी बार बार अपॉर्चुनिटी क्रॉप्स की पोटेंशियल क्रॉप्स की बात हुई, तो इसमें एक अच्छी प्रगति मेरे विचार से पिछले काफी समय से हम कर रहे हैं। और उसमें जो है लगभग 21 वैरायटीज पिछले 4-5 साल में हम नोटिफाई कर पाए, और उसको अपस्किलिंग को भी मेन स्ट्रीम को भी काफी मात्रा में कार्य हम कर रहे हैं। अन्य एक्टिविटीज में जिस तरह से मैंने बताया कि आईटीजी प्यारे की जो एक मीटिंग हुई, तो उसमें मैं और डॉक्टर उसमें थे, तो जितना भी एक्सपेंडिंग ऑन एमएलएस और स्टेटस ऑफ बीएसआई बेनिफिट शेयरिंग आदि पर चर्चा हुई, और हमने अपना पूरा जो देश के हित में जो भी बातें हुई उस पर हमने वहाँ पे कार्य किया। जो जॉइंट सेक्रेटरी सीट भी साथ थे। ब्रिक्स की मीटिंग में डीएफडब्ल्यू की विभिन्न पॉलिसी इश्यूज में, मिनिस्ट्री ऑफ एनवायरमेंट, फॉरेस्ट, क्लाइमेट चेंज में, एनबीए और अन्य गतिविधियों में हम लगातार उस पर कार्य करते रहे और आगे भी हम कार्य करते रहेंगे। जो जर्मप्लांट डाइवर्सिटी डे जो कि एक्टिविटी है, जो वालों के वैज्ञानिकों को, संस्थान के वैज्ञानिकों को बुलाकर और उसको जो है जो फील्ड डे कराना, और जिस तरह से भी मटेरियल को सेलेक्ट करते हैं तो उस मटेरियल को हम बाद में उनको भेजते हैं, और ये कार्य भी सुचारू रूप से करते हैं। ट्रेनिंग प्रोग्राम्स का, जैसे कि हमारे पास एनईएस, टीएसपी, एससीएससी प्लान में काफी पैसा रहता है, तो उसमें 30 कार्यक्रम पिछले साल किए गए, जिसमें लगभग 5,000 जो फार्मर्स बेनिफिशियरी इन कम्युनिटीज के रहे और उनको ट्रेनिंग का कार्य किया गया। संस्थान के बहुत से वैज्ञानिकों को इसमें इस वर्ष में काफी अवार्ड रिकग्निशन मिले, तो इसके लिए भी मैं सभी को बधाई देता हूँ। यहाँ तक जो 6 ट्रेनिंग साइंटिस्ट की जिसमें 34 वैज्ञानिक हमारे भी बाहर ट्रेनिंग के लिए गए, और जो पीजी स्कूल की एक्टिविटीज में 11 स्टूडेंट्स हमारे इस बार पीएचडी में, टोटल 6 पीएचडी में और 5 एमएससी में एडमिट हुए, और जिसमें 7 स्टूडेंट्स ने 4 पीएचडी और 3 एमएससी में अपनी डिग्री कंप्लीट की। पूरे में कुछ जो कंस्टेंट्स भी हैं, क्योंकि डीजी साहब भी है, उस दिन भी चर्चा हुई थी, टेक्निकल स्टाफ की एक बहुत भारी कमी है, और सभी अध्यक्ष और जितने यूनिट इंचार्ज की वैज्ञानिकों की इशू रहता है। अभी 38 टोटल 108 स्ट्रेंथ में से 38 टेक्निकल स्टाफ हमारे पास उपलब्ध है, और टोटल वैज्ञानिकों की संख्या इन प्लेस है वो 112 है। और अभी पिछले 1-1.5 महीने में 5 टेक्निकल स्टाफ का ट्रांसफर हुआ, जिसके लिए कि मैं ये निदेशक, मेरे को बड़ी चिंता रहती है, क्योंकि रोज मेरे को वैज्ञानिकों को फेस करना होता है। इसी तरह एडमिन स्टाफ में भी 40-86 में से आज की डेट में हमारे पास है। और अगर बात करते हैं तो 5 डिवीजन है, 4 यूनिट्स हैं, एक एआईएनपी है, सीआरपीएन एग्रो बायोडाइवर्सिटी है, और उसके अलावा जो 10 रीजनल स्टेशन, और जो इस तरह का जो काम है 50 के आसपास जो एक्सटर्नली फंडेड प्रोजेक्ट्स हैं। तो काम को देखते हुए जो एडमिन स्टाफ की भी काफी कमी है, और खास तौर पे असिस्टेंट आदि पीछे कुछ आए थे, तो मेरे विचार से जो जितने आए थे उससे ज्यादा लोग ट्रांसफर करके चले गए, तो ये एक बड़ी समस्या है। सपोर्टिंग स्टाफ की टोटल पोजीशन 63 है और आज की डेट में 34 है, और इससे जितनी भी कॉन्ट्रेक्चुअल मैन पावर लेते हैं तो हमारा ज्यादातर पैसा है जो वो कॉन्ट्रेक्चुअल मैन पावर पर चला जाता है, क्योंकि वो सब कंटीजेंसी अमाउंट से उठते हैं। तो वे फॉरवर्ड की बात करते हैं, परसों की 29 तारीख की मीटिंग में भी ये चर्चा हुई, और मेरे विचार से हमारा डेडिकेशन ज्यादा रिसर्च का, प्लांट जेनेटिक रिसोर्सेज से रिलेटेड रिसर्च पर होना चाहिए, ना कि हम इधर उधर की उसमें एक्टिविटीज में इन्वॉल्व हों। एक जो आज की जो हम बात करते हैं एनवायरमेंट क्लाइमेट चेंज की, तो जितने भी नए इंसेक्ट पेस्ट हैं, नई डिजीज हैं, तो वो एक फूड सिक्योरिटी को बड़ा खतरा है। तो उस तरफ को ध्यान देके, चाहे हमारे कोई भी डिवीजन हो, और जीनोमिक रिसोर्सेज डिवीजन की एक विशेष तौर पर जिम्मेवारी बनती है कि आज हम जो भी जर्म प्लान हम एक्सचेंज करते हैं, तो उसमें कम से कम एक इंडिपेंडेंट डिवीजन होने के नाते उसमें जो भी हम जर्म प्लाग दें, अगर वो ट्रेट स्पेसिफिक जर्म प्लाग है और उसकी जीन सीक्वेंस भी साथ अगर देते हैं उस पर्टिकुलर ट्रेट की, और जो मार्कर देते हैं, तो दैट विल बी गुड, आई थिंक, हेल्प टू द ब्रीडर्स, वेयर वी कैन एक्सपेडाइट आवर एक्टिविटीज rather than going and then having innovation, then go for the trade, so that is essentially required. कम्युनिटी सीड बैंक पे अच्छा कार्य शुरू किया, इस पर कार्य करने की आवश्यकता है। और जितना भी हेरिटेज साइट्स का भी कुछ संस्थान ने काम किया, जिसमें के लास्ट ईयर जो जैसे उसमें हमने एक जो बरजुल्ली में जो वाइल्ड राइस के लिए एक हेरिटेज साइट सोनितपुर आसाम में एक आइडेंटिफाई की, हमारा योगदान रहा। इसी तरह हैबिटेट ऑफ कजनस कजन पोलियस के जो फॉरेस्ट है, इसमें बेलडेलिया रेंज इन दंतेवाड़ा में छत्तीसगढ़ में, और गुआवा का जो है एक थाल पिथौरागढ़ यूके में जो उत्तराखंड में, तो वो उसकी हैबिटेट वेरिएबिलिटी को कंजर्व करने की कोशिश की। तो इसी तरह का और कार्य हमें जो है करने की आवश्यकता है। प्री ब्रीडिंग पे बहुत जोर रहा, और क्योंकि आज की डेट में हम देखते हैं तो जर्म प्लाग का जो यूज है, उस दिन चर्चा हुई कि 1% से कम हम टोटल जर्म प्लाग में हमारे पास है उसका कर पा रहे हैं, उसके लिए हमें और उसकी मात्रा बढ़ाने की आवश्यकता है। और प्री बिडिंग की एक्टिविटी ब्यूरो में जब वैज्ञानिकों की संख्या को देखते हैं तो मेरे विचार से जो क्रॉप इम्प्रूवमेंट से रिलेटेड पर्टिकुलरली प्लांट ब्रीडिंग जेनेटिक्स के वैज्ञानिक हैं, तो उनको इस कार्य की तरफ आगे मैं अभी कुछ व्यवस्था करूंगा कि प्री बिडिंग का कार्य हम अभी कर रहे हैं, लेकिन उसको और आगे बढ़ाने की आवश्यकता है। सेफ्टी कॉपी जीन बैंक एक बड़ी उपलब्धि रही, और इस पे जो बजट अनाउंसमेंट 2025-26 का था, तो हमने पहले से उस पर कार्य शुरू कर रखा है, और डी साहब की उसमें मार्गदर्शन में हम बहुत ही जल्दी जो आईसीआर का 100 ईयर्स का फाउंडेशन डे है, उस समय तक हम कोशिश करेंगे कि हम सेफ्टी जीन बैंक को जो सतिनगिरी में किलोंग हिमाचल प्रदेश में उस पर कार्य हम कर रहे हैं, तो उसको जल्दी से जल्दी कंजर्वेशन डिवीजन की मदद से तो उसको हम एस्टेब्लिश करने की कोशिश करेंगे। जो रैपिड डिटेक्शन टूल की बात करते हैं क्वारंटाइन में खास तौर पे, और उसमें निश्चित रूप से हमें उस तरफ भी जीनोमिक्स डिवीजन और क्वारंटाइन डिवीजन दोनों मिलकर काम करें कि जितना भी एक फास्टर सिस्टम हमारे पास हो, अदर देन हम उसमें ग्रीन हाउस में लगाते हैं, पॉली हाउस में लगाते हैं, सीजन की वेट करते हैं, तो इस तरह का कुछ सिस्टम हम शुरू करें कि जैसे ही कंसाइनमेंट आए हम उसको तुरंत जो है जो भी इंडेंट है उस तक पहुंचाने में हम कामयाब हों। जितना भी जर्म प्लान जाता है और उसका जब कभी भी उसका उपयोग की बात करते हैं तो हमारी स्थिति बड़ी निराशाजनक रहती है, और कहीं भी कोई पूछता है कि आपके जर्म प्लान से कितनी वैरायटी आई, तो उसके लिए हम हमेशा निरुत्तर रहते हैं। तो मेरे विचार से जो कंसर्न डिवीजन है इसमें एक ट्रेसेबिलिटी का सिस्टम हमें शुरू करना चाहिए कि जितना भी जर्म प्लान हमारा जाए उस पर हम कार्य करें, और उससे ताकि जो ब्यूरो का एक मेजर कंट्रीब्यूशन हम बता सकते हैं कि जो हमने जो मेहनत की जर्म प्लान कलेक्शन, करेक्टराइजेशन, कंजर्वेशन, और उसके बाद जो लेके गए जितने भी संस्थान उसका ये यूज हुआ, और इस तरह की वैरायटी जो उसमें आई उस पर भी कार्य करने की आवश्यकता है। We all feel proud of the very significant progress made in the past and during the current year in terms of all the areas like the varieties, technologies, and the other patents and the conservation as well as exchange of the material. And this Foundation Day, you all are well aware that it is not only to get together and having some lectures and then go ahead, it is a day of introspection. What we have done during the past 50 years and what we have done during last year, and where we have to grow, what are the priorities of the government of India. And when we talk of the Viksit Bharat during 2047, so the role of Bureau is very important. So I think we need to think over that, and we need to work more and more systematically. On the occasion of this 51st Foundation Day of the Institute, I congratulate all the scientific, technical, administrative, finance, supporting staff, students, contractual personnel of the institute, and also remember the contribution of our previous managers and all the staff. NBPGR has always the priority of the ICR also, whether it was in the time of Dr. Mahapatra, Dr. Pathak, or now our Dr. M.L. Jad. So there is no dearth of financial resources, which I observed during last my 9.5 years experience at the council. So we sanctioned last time the quarantine facility around 1.5 crores for the DPQ division. And under the new mission which has been launched during this month is Global National Mission on Housing Seeds, which I am looking after at the national level. In that also a very mega facility like DNA fingerprinting laboratory, a state-of-art central laboratory, which will be the, I think, notified by the Ministry of Agriculture also. And also some of these quarantine facilities will also be established. And thanks to our DG Saab, our former DGs and the DGs of Crop Science Division, our DGs, and my sincere thanks and gratitude to Dr. R.S. Pradha Saab for his guidance and support all the times. And my colleagues from the council, I forgot Dr. Partho and Dr. Prashant, who are there. So their help always for the division, I think, for the institute. So thank you. You also, our Honorable Secretary ICR. So he has joined 2 months back, but his involvement and interest and support in various activities, sir, I am thankful to you, sir. My sincere thanks to all our former directors, all the staff who have joined physically and online, and all the directors of our sister institutes who have joined online. So my sincere thanks to all of you. My sincere thanks to whole INBPGR family for their support and hard work in making the program a grand success. I wish you all the best for a very successful time ahead. Thank you. Jai Hind. Namaskar.
धन्यवाद सर। पिछले वर्ष के दौरान ब्यूरो की महत्वपूर्ण उपलब्धियों एवं उल्लेखनीय प्रगति का विस्तृत विवरण प्रस्तुत करने के लिए आपका हार्दिक आभार। इसी क्रम को आगे बढ़ाते हुए अब मैं श्री ज्ञानेंद्र देव त्रिपाठी जी, एडिशनल सेक्रेटरी डेयर एंड सेक्रेटरी आईसीआर से विनम्र अनुरोध करती हूँ कि कृपया मंच पर पधार कर अपना संबोधन प्रदान करें।
Namaskar, good morning, and a very happy time. Very distinguished personalities who are on dais, particularly Dr. R.S. Paroda, Chairman TAS, and former Director of ICAR. He was also Director General of ICAR, and he was also founding director of this institution. So, welcome. In fact, it's a great opportunity to see Dr. Paroda here. Dr. Jat, Secretary DARE and Director General of ICAR. In fact, he is, I can say, for all purposes, my mentor since I have joined in the ICAR. In fact, every day, every passing minute, I'm learning a point or two whenever I meet or interact with sir from time to time. So grateful to you, sir. Then Dr. Nagendra Kumar Singh, he is J.C. Bose National Fellow and former National Professor of B.P. Paul Chair, ICR NIPB New Delhi. So welcome Dr. Nagendra Kumar Singh. And Dr. R.K. Gautam, who is also on the dais, very distinguished scientists, very distinguished officers of National Bureau on Plant Genetic Resources, and distinguished personalities who have joined online, and ladies and gentlemen. So today, when we are on the 51st birthday of this institution, 50 years we have already passed by, and we have the huge celebrations in our mind that we have 4,75,000 accesses, which is nothing but second best in the world. Can we become the best in the world as we celebrate some other occasion, maybe in 2, 3, 4, 5 years, whatever it is. So here comes one opportunity. In fact, this second copy gene bank, which Dr. Yadav mentioned in his welcome address, that is going to be a game-changing the infrastructure and game-changing storage for the genes. It will be a kind of disaster recovery site and being set up at a very, very nice location. So I think there is a time to commit that when after 2 years we are going to celebrate this 100 years of ICR. So if not just on that 100th birthday, at least in the 100th year, we should be in a position to dedicate that particular second copy of Gene Bank to the nation at some point of time. So this is a commitment which we must take today. And in fact, when it recently I was in the Northeast, I went to Shillong, and there I had opportunity to discuss and go to some community gene bank, community seed banks. So there we saw that seeds are stored in a very, very pristine way, and their pristine methods of preservation. In fact, one of the Khasi ladies, she came forward and she said कि हम लोगों के यहाँ पर ये दादी माँ का बीज के नाम से जाना जाता है। यहाँ पर वो उन लोगों ने अपने हिसाब से बोला, लेकिन उसका ट्रांसलेशन इस तरह कि दादी माँ का बीज होता है, और इसको हम लोग बहुत जन्म जन्मांतर पीढ़ी दर पीढ़ी जो है प्रिजर्व करके रखते हैं। और वो कुछ नहीं, छोटी सी एक मटके टाइप का था, उसमें बीज रखा हुआ और कुछ राख रखी हुई है, इस तरह से जो है। तो ये है ट्रेडिशनल कम्युनिटी प्रिजर्वेशन. एंड व्हेन डॉक्टर यादव said that out of 4,75,000, more than 1 lakh are the traditional indigenous varieties. That is basically the success of this particular the NGPGR, that stores not only the exotic varieties but also the varieties which are very own and which can become a kind of tool. So one day I asked one scientist that why so much preservation, because these are preserved at some place anyway. So then he said, no, sir, actually the problem comes not in the normal preservation or in normal times. There can be a situation when suddenly a disaster comes in the form of a new disease or new kind of attack on the crops. And then some old seed which is kept in our records, and that may provide that a particular gene that is resilient to that particular attack or that particular disease, and that helps in a big way. So it is not only a living seed, but it is also a kind of library to refer back to that knowledge which is preserved by our people, our ancestors, and our communities. So with this, I think, objective in mind, and the Viksit Bharat the goal which is already in front of us, and the second copy of Gene Bank which is already in the making, I would like to wish very, very best to all of you on this Foundation Day, which is 51st. And maybe when we meet after few Foundation Days, we can say that we are the globally best gene bank in the world. Thank you so very much.
धन्यवाद सर। इसी क्रम को आगे बढ़ाते हुए अब मैं जर्मप्लाज्म मूल्यांकन प्रभाग के विभागाध्यक्ष डॉक्टर आर.के. गौतम जी से विनम्र अनुरोध करती हूं कि वे आज के 13वें एच.बी. सिंह स्मृति व्याख्यान के विशिष्ट वक्ता डॉक्टर नागेंद्र कुमार सिंह जी का परिचय प्रस्तुत करें। आज के व्याख्यान का विषय है आइडेंटिफिकेशन एंड यूटिलाइजेशन ऑफ QTLs फ्रॉम क्रॉप जेनेटिक रिसोर्सेज। डॉक्टर गौतम, प्लीज।
Sadar Namaskar. Good morning. Honorable Chief Guest of the Golden Jubilee Program, Padam Bhushan Dr. R.S. Parodaji, Chairman TAS, former Secretary DARE and Director General ICR, and former Director ICAR-NBPGR. Honorable Chairman of today's function, Dr. Mangilal Jatji, Secretary DARE and Director General ICR. Most respected Dr. Shri Gyanendra Dev Tripathi Ji, Additional Secretary DARE and Secretary ICR. Respected Dr. Devendra Kumar Yadav Ji, Deputy Director General Crop Science ICR and Director ICAR-NBPGR. Respected Professor Nagendra Kumar Singh Ji, J.C. Bose, a National Fellow and former National Professor ICAR-NIPB. All the distinguished guests, staff members of ICAR-NBPGR, press and media, ladies and gentlemen. We are all now eagerly waiting for very momentous occasion, and especially for the most awaited 13th Dr. Harbhajan Singh Memorial Lecture by a highly accomplished and distinguished scientist, Professor Nagendra Kumar Singh Ji. It's my great privilege and honor to introduce our distinguished speaker, Professor N.K. Singh, one of India's most eminent plant genomics scientists. Professor Singh is currently a J.C. Bose National Fellow and is internationally recognized for leading the decoding of the genomes of several economically important crops, including rice, tomato, wheat, pigeon pea, jute, and mango. His pioneering work has significantly advanced plant genomics and crop improvement in India. He completed his post-graduation in genetics and plant breeding from Banaras Hindu University, Varanasi, followed by his PhD from the University of Adelaide, Australia. Throughout his distinguished career, he has served at CSIR, CFTRI, Mysuru, G.V. Pant University of Agriculture and Technology, Pantnagar, and later at ICR, National Institute of Plant Biotechnology, IRI, New Delhi, where he made landmark contributions to plant biotechnology and genomics, and especially for leading the genomics program in the country. Professor Singh is the founding president of the Genomics Foundation, New Delhi, where the genome of the iconic flame of the forest, Plas tree, has been decoded. Under different genomics projects, including QTL2 variety project, he has led the development and commercial release of 22 climate-resilient rice varieties and 6 dwarf pigeon pea varieties for mechanized cultivation. This amply exemplifies the concept of gene discovery to variety and seed delivery. His contributions to the conservation and utilization of wild rice genetic resources have resulted in the discovery of valuable genes for abiotic stress tolerance. During his research in Australia, he discovered a new class of wheat storage protein called as trypsin, which has had a lasting impact on wheat quality research. He has mentored more than 100 postgraduate students, published over 400 peer-reviewed research papers, and has received more than 37,000 Google Scholar citations with an impressive H-index of 88. Today, we are fortunate to have him share his expertise on the topic identification and utilization of QTL from crop genetic resources. Honorable dignitaries, esteemed audience, ladies and gentlemen, please join me in extending a warm welcome and give a big round of applause to Professor N.K. Singh. Sir, I humbly invite you to deliver the keynote lecture. Sir, the stage is yours. Thank you, sir.
नमस्कार, जय हिंद। आदरणीय डॉक्टर परोदा साहब, आदरणीय डॉक्टर एम एल जाट साहब, आदरणीय श्री ज्ञानेंद्र चतुर्वेदी जी, डॉक्टर डी के यादव सर, डॉक्टर आर के गौतम, and dignitaries in the hall and also who have joined online. First of all, my humble regards to all of you, and very, very highly congratulating you to this 51st day of your foundation day and Golden Jubilee celebrations. To all of you, all the scientists and all those who are associated with the NBPGR. At the outset, I would like to pay my humble tribute to Dr. Harbhajan Singh ji, to the memory of Dr. Harbhajan Singh, who has been the visionary to start this program of the plant genetic resources in India, which has grown up to this level of international standard institute, which Dr. D.K. Yadav has described in detail, and also you have heard from the Adani Secretary. So today I would like to thank Dr. Yadav for inviting me for this very prestigious Dr. Harbhajan Singh Memorial Lecture. So it is my privilege, sir, to join this lecture, deliver for you. The time is short, so I will go straight to the lecture. Although I have been part of the last QRT of NBPGR under the chairmanship of Professor Dhillon, and we have been through, to few months, the entire activities of the institutes, and who has done exceptionally well actually, and it is growing, day by day to reaching the new heights. And already you are the second biggest germplasm bank, and I hope and I wish and I'm sure also that one day you will be beating the United States, because we are very rich in the biodiversity of the plant here in India, and we can soon, I think within less than 5 years, we will be more than USDA, and that is very, very important. So my talk is on the identification and utilization of the genetic resources for the QTLs and genes which can be utilized in the crop improvement, because the purpose of this changing. Yes, it changed. So you can change for me. PDF should also change. Somebody can change it for me. Yeah, I also change. Somebody has it. Who change it? Okay. So at the outset, sir, I would like to start with the acknowledgements, because sometimes we forget towards the end. So mostly I will be describing the work done by our own group, some work from outside. And all this work would not have been possible without the support from the system. And Dr. Paroda Sahab is the biggest mentor for me also, who got me here from Pantnagar to start the Rice Genome project. And we have received a very large funding from ICR and DBT. I will not go into detail, but I will just mention some of them. The NPTC project has been really a plan-based support, Indo-US Knowledge Initiative of the ICR, and the National Professor Project, Norman Borlaug Award Project. There's a huge funding has been there. And then from the Department of Biotechnology, they have funded in a big way the big genome projects on rice, wheat, and tomato. The other projects have been funded by ICR. And then the translational projects like UTL to variety project and the Indo-Swiss collaboration in the biotechnology of Pigeon Pea, which led to the development of dwarf varieties. So I would like to acknowledge their support. Large number of scientists have been associated who worked with me. I'm not going to read individual names because the list is very large, and still larger number of students and the project staff. So it is not the work of a single person. There are many people have contributed what I'm going to present here. This is the outline of my presentation. We need to use the modern tools, and you already have a division of genomic resources here, which helps actually. So I will just give a little bit of introduction that how this is helping and how the genome sequencing has been done very, very briefly. Then gene discovery and utilization pipeline, how we go from the genetic resources to the final product, what is the pipeline there and the timeline. Then one example of this QTL to variety project where large number of climate resilient varieties have been released. Then I will go through the search for new genes in the wild rice germplasm from exploration till the identification of the genes. And then simultaneously mapping and interrogation from the genetic resources. Then some of the examples from some other crops. And finally, the challenges and the way forward. So I think most of you know that the genome is defined as the complete set of genetic information present in a single haploid cell of an organism. This definition was given more than 100 years back and it still holds true, including the chloroplast and mitochondria. So what is the use of genome? Why we spend so much money and effort to do the genome sequencing and annotation? Once we have the genome, which is the physical map of the genome rather than the genetic map. So base by base, each base makes this physical map of the genome. Then we can do the species comparison, how different species are correlated with each other, how far back they had common ancestors, and how close or far they are from each other. That is very important for the curiosity of knowledge and also for choosing the parents in the breeding program. Then coming down the list is the practical application. High density DNA fingerprinting is very much essential for protecting our germplasm and the IPR property, because low density fingerprint can be easily bred out from the material and you will not be able to prove that it is your material. Then genome information works like a GPS, like you have mobile GPS, you can get to the people, to the places. So having genome is like a GPS for gene discovery. You can identify the genomic regions and eventually the gene which are responsible for the trait, and that is needed for the crop improvement. Then actually the breeding process itself, like marker-assisted breeding through foreground and background selection, genome information is very crucial. And finally, the last in the list is the genome editing. It is impossible to do genome editing without having the genome information, because when you develop your guide RNA, you have to ensure that there is no off-target effect. So unless you have the complete genome, you cannot be sure that there is no off-target effect. Now that's how it all started when we joined from the Pantnagar year. Dr. Paroda Sahab was DG at that time. So we got big funding from DBT, although it was a joint project between ICR and DBT, but the funding came from the DBT after a lot of discussion. And then, to cut the long story short, within the ahead of the schedule, we completed the work, and it was declared Japan was the main contributor, and we established everything. We learned the techniques right from the sketch and published this paper in Nature, which has been cited more than 5,000 times because it is very useful to people worldwide. They are using this information for gene discovery. But the way we did was very hard way to do because each piece of DNA has to be cloned and multiplied in bacteria with the toothpick, clone by clone, 2 kb at a time. And that was really a long, winding road. And that is in the hindsight because in 2008, when this NGS technology came, which we are using nowadays, we are not doing the sequencing by cloning now. So this technique came, and that is speeded up the sequencing. So the tomato genome sequence was, initially it was planned to sequence only the gene-rich region, but with the new technological innovation, now complete genomes were sequenced. And similarly, it was also published and being used every day. And then wheat genome was also completed using the same methodology of the NGS technology. So they are very big genomes were completed, and we were partner through the big support from the DBT and ICR. Just in 2015, the latest technology came where you don't need to clone, do not to PCR amplify, simply take the DNA and original DNA can be sequenced. By single molecule DNA sequencing, you get long read lengths. So now sequencing has become very easy now and very cheaper, not as expensive and as hard as it was in 2000 and 2005. So now a lot of genomes are getting sequenced, and we ourselves have sequenced so many other genomes, like this, the Kalanamak rice. We finished it to the telomere to telomere. This sequence is better than the Japonka sequence, which was done internationally because the technique has improved. And then this I want to focus on this one. Here the gene discovery and utilization process flow and pipeline. In fact, the pointers does not work on the electronic screens, but that the first circle on the left side is the stakeholders' preferences. We are all working for the society. And the last one is that what we give to the society, that is varieties. The whole plant breeding, genetic resources, all the basic disciplines, their work for the society is that they take the what society needs and eventually give you the package of practices and the varieties which society can utilize. And then the feedback will come. With the climate change and the changing lifestyles, mechanization, the need will be changing, and all the stakeholders will tell you that what they need, and we as a scientist, we will start improving the process of the natural resource management as well as the variety. Those are two sides of the thing. But then everything starts from the genetic resources. The second circle is the genetic resource. The gene, where are the genes? The genes are in the germplasm. That is the wild relatives, traditional varieties, and the varieties. Without those resources, so that is the key resource for working towards the improvement of the varieties. And there are a lot of different techniques, high throughput techniques for the germplasm evaluation, and we can identify the donors of the traits. And then in the second step, we can do gene discovery through genomics assisted techniques like functional genomics, QTL mapping, fine mapping, expression profiling. I'm not going into detail, I'm talking more generic here, that using a lot of genomic techniques and the traditional disciplines like biochemistry, physiology, and also field evaluation proper, we can identify the genomic regions and also the genes which are responsible for the trait improvement. And then we can go allele mining. We can go back to the germplasm. Each gene has several alleles, several alternative forms, and we can fish out the best farm, the most desirable farm, and then used through marker-assisted breeding. So through this science-based approach, technology-based approach, we can speed up the breeding process. From traditionally it used to take 10 to 15 years to release a new variety from when you made the first cross. In the marker-assisted breeding, within 5 years, a variety has been released and has been released. Number of varieties, because now you have the entire information about the background, the foreground, and the gene you want to transfer. So this cycle is actually a generic kind of things. How from the society's basis, based on their preferences, we can look for the genes in the germplasm resources, identify the genes and markers, and then through breeding, we can deliver the product. Now, this is another way I want to depict here that why germplasm conservation is so important. You can see the wild progenitor species, say for example rice, it was domesticated from a wild rice, Oraja rupipogan and Nivara. So that progenitor species is very rich in the diversity. It has more number of genes which are represented here by different shapes, like six genes with 6 different shapes and 4 colors for 4 different alleles. So the progenitor species has more genes and more alleles. Then when traditional farmers, they domesticated it, they just pick few of them. So that few representative which make the crop species, they have less number of genes and less number of alleles also. And when you come to the modern, highly developed green revolution varieties representing the monoculture, they not only have the fewer number of genes, but only one allele. Each variety is pure. So we have left behind, we have not lost, we have left behind number of genes and alleles which will be very useful. And the germplasm which we are conserving, basically we are conserving that. So we have to go back to that germplasm and find more genes and more alleles useful for the need of the group. Now I will give you we targeted only 3 traits. So first we discovered the trait, we find a trait in a germplasm. Then using the genomic techniques, we can identify the genes. So here are the 3 traits like high grade number, exceptional quality of basmati rice, and the salt tolerant. Now, none of these are variety. They have the trait, but they are not good enough to be called as a variety. So if I take the gene from here and put in the highly productive background, then that becomes an improved variety. So I will give you just only one example, that the trait for high grade number, it took 10 years from the mapping, fine mapping, expression profiling, and maybe number of students. Now we got to the gene. On the long arm of chromosome 4, there is a gene for kinase. When it is expressed in higher amount, and that increases the grain numbers. And eventually it increases the yield also. There is no compensatory effect. So it was transferred in 12 mega varieties, which are already ruling varieties for last 40 years. We transferred them in all of them through foreground marker. And then for background selection, so ensure that the original varieties, all the other properties are recovered, we developed a chip, high density chip, that is indigenous chips were developed, 50K SMP chip. And through foreground and background selection, see background is blue, foreground is red. So in a best of the best varieties, we transfer one gene per yield, and then the yield was increased. So we say that yield is quantitative traits, it is very difficult to work with, but you can have major genes per yield also. So here is in the Samba Masuri background, the pancreas size was doubled, well-filled grain. The tiller number was slightly reduced, but the overall 15 to 20% higher productivity. Similarly, in Pusa 44, also in 12 mega varieties, we transfer this, and they are in the EBT. I don't want to make negative comments about the ECRIP, but this variety rejected from ECRIP, but it has found its way from the Karnataka, it is getting released. So when it is productive thing is there, it will find its way. If your material is good, it will find its way. Similarly, we'll identify the genes for the basmati quality traits, which we are ensuring. Whenever you're doing cross breeding, you have to get back your original quality by back crossing. So this is the example of climate proofing of mega varieties of rice. So these mega varieties of rice, basically they are the rare combination of genes and alleles. Whether we like it or not, accept it or not, your best varieties in rice and many other crops are those which were 30 years, 40 years old. And still they are ruling. MTU 10 has the highest breeder seed indent of rice. Even if you do notify, don't give subsidy, still farmers are using because it is best for them. So we have to accept it. What is best, we have to accept. Unless we become better than that, farmers will not take it by whatever rule we pass. In other thing, but these varieties have defect. They are sensitive to environmental stresses, climate change, diseases, and sometimes they don't have the same quality as the traditional varieties. So traditional variety is resilient to climate. It has good, excellent quality, but still people will not grow it. We partially, lie the blame on Green Revolution that we lost so many varieties. Every variety is there in the gene bank, as Dr. Yadav pointed out. Even if I go give it to farmer, he will not grow it for the same reason why he discontinued growing it. But what we can do as a scientist, we can take out the genes for climate resilience and the quality. Everything genes can be identified and then put it back in the high yielding background, then they will take it. So there are different methods of improving the variety through marker-assisted breeding, but the marker-assisted backcross breeding has been the most successful to date. I can just, sir, here I want to point out one aspect which is, which I understood after 35 years of being a student of genetics and plant breeding. It is simple Mendelian formula that in the F2, if you want to have the better allele of the two genes only, the probability is 1 in 16. It is 1 over 4 h to the power n. If the number of genes increases, the probability will also decrease. So if you want to have the 10 genes, here I have given 12 genes, one gene on each chromosome of rice. You cross it, then in the progeny, you want to select a plant which will have the better of the 12 alleles for each of the 12 genes. The probability is 1 in 16 million. If you don't believe me, you can ask AI or Google. A simple math is there. Your 4 gene, 1 in 256, 10 gene is 1 in million, and you keep on multiplying with 4 for every additional gene. So these mega varieties are actually the cream of the 100 years of breeding work. And that's why it is not easy to surpass them. Because when we cross to something and then you want to again put 15 genes together, so is your breeding population big enough to allow that to happen? Because there is no shortcut to that, except for if you want to do genome editing, put 20 genes together and make it a one gene, behave like one gene and put them together. So otherwise it is not possible. So this is the real challenge we have to keep in mind. And that's why to improve upon gene by gene, the best variety, just keep on adding genes to that. And that is the simplest strategy. So we set out to add genes for climate resilience, drought, flooding, and salinity. And DBT funded to the tune of 27 crores in 10 years in 2 phases. 17 crore in the first phase, second phase 10 crore, and 15 institutions were involved. Second phase में हमने 3 ड्राफ्ट कर दिया तो 12 थे. And it was a very well focused program to improve the mega variety of rice. Just 3 backcasts दे करके background select कर लिया, additional gene डाल दिया. Now it is replacing the old variety. Now it is replacing. When by MTU 1010 में हमने 2 gene डाल दिया salt tolerance के लिए, पूरा कोस्टल आंध्र प्रदेश में it is replacing like a wildfire. रंजीत में सब डाल दिया, आसाम में उसने replace कर दिया. But any new bred variety have not been able to replace. Same the golden variety में अगर 2-4 gene और डाल दिया useful, तो वो replace करेगा. For the reason I have told. So we standardize the markers, foreground, background. This is the list of 19 variety. 3 more have been added. So 22 variety from a single project, and all of them getting seed indent. It has reached into millions of hectares already. And the last 2 साल का जो seed indent है वो अगले 2 साल में फील्ड में पहुंचेगा. So there are some examples like here are 50. Just this year 20 quintal indent was obtained from Chhattisgarh. So even if there are no mentor for this variety, if the breeders has retired and it is not favored by the current dispensation, still because it is good, farmers are demanding the seeds of that. Then MTU 1293, it has got 2 genes per salt tolerant. Already the best indented variety, 2 more genes makes it unbeatable. Then there is a HUR 105. Similarly, there are more varieties. So I just gave you the examples of that, how the mega varieties can be improved by adding what is needed and then back crossing to recover what was already there to make it best. Now the genes are in the germ plan. Germ plan, we have to go back. Dr. B.P. Pal has a slogan of search for new genes. New genes के लिए germ plan होना चाहिए. When I started National Professor Project, only 200 wild rice collections were there. Now more have been collected. Dr. Yadav, Dr. Halawat, they have done several expeditions. So I collected personally. I went to several states and villages. Wherever it is present in abundance in the roadside, in the shallow ponds, in the river basins, in the small ponds. Then there are some rare sites like in the hill from Himachal to Karnataka. Even a small pit will have wild rice. It will grow, it will settle, it will, from millions of years it is there. It has survived all the global warming and global freezing. Then it survives, it is adapted to the high soil condition, coastal saline, drought, flooding. It must have genes for that. That's why it has survived. Then most important thing is that it is threatened. It has survived so far, but it cannot bear the brunt of development. You can see, you cannot find any wild rice in the western UP, Haryana, Punjab. जितना डेवलप्ड है उतना बायोडाइवर्सिटी लॉस हो गया है. सो आप ईस्टर्न स्टेट में फाइंड कर सकते हैं, वहां पर भी घर बन रहा है, स्कूल बन रहा है, इंडस्ट्रियल पार्क बन रहा है, रोड चौड़ा हो रहा है. एंड ऑल दीज डेवलपमेंटल एक्टिविटीज we are losing our diversity at an alarming rate. What has evolved through millions of years in one stroke of development, mindless development sometimes. रोड को चौड़ा करना है तो एक साइड में करते तो आधा बच जाता है एक साइड का. दोनों साइड में करके बड़े बड़े आम के पेड़ सारा डाइवर्सिटी खत्म कर दिया. नाउ यूपी लुक्स लाइक राजस्थान इन दिस समर. Whereas you used to have big tree of mango, जामुन, ये सब रोड के दोनों तरफ थे. दोनों तरफ का पहले काट दिया, फिर रोड को चौड़ा किया. Not only in UP, this figure is from Andhra Pradesh, from Karnataka. पूरे कंट्री में यही हाल है. जहाँ डेवलपमेंट हुआ आपकी डाइवर्सिटी गई. तो इसके लिए हमें कुछ करना पड़ेगा. कंजर्वेशन पार्क एक्सेट्रा जो भी वेटलैंड्स हैं उनको प्रोटेक्टेड घोषित करना. वी हैड लॉट ऑफ डिस्कशन एंड देन डॉक्टर अलावत मेड एफर्ट. NBPGR आसाम में एक डिक्लेयर हुआ है, लेकिन यूपी में लाइक बलिया में सुरहा ताल है, आजमगढ़ में गंभीर बंद ताल है, वहां पे बचा हुआ है. लेकिन इफ इट इज नॉट डेवलप्ड, सुरहा ताल में चंद्रशेखर यूनिवर्सिटी बन गई, गंभीर बन में अटल रेजिडेंशियल विद्यालय बन गया. एज इफ देयर इज नो प्लेस फॉर डेवलपमेंट. यहीं जहां पर थोड़ा बहुत बचा हुआ वहीं पर डेवलपमेंट भी हो रहा है. एंड एंड रिजल्ट यू हैव सीन. नाउ वी कलेक्टेड अबाउट 1000 एंड इट इज इक्विवेलेंट टू 10,000 लैंड रेसेस, क्योंकि वो डाइवर्सिटी 10 times मोर है वाइल्ड राइस में. वी हैव डन सम साइंटिफिक वर्क हियर एंड प्रूव दैट द rice was domesticated in India, not in China, because with the help of technology they were misleading the word that all rice domesticated in China and then वहीं से पूरा दुनिया में गया है. But once you prove, do this scientifically, you can prove it that हमारा आउस और इंडिका यहीं पर इवॉल्व हुआ है, क्योंकि it is closely related to our germ plant. Then when I screened it, we find better than the cultivated for drought tolerance also, for anaerobic germination, for salt tolerance, for BLB, for any trait, for sheath blight जिसको cultivated में source नहीं है, you can see wild में है. Then brown spot disease. Then nematode. For direct seeded rice, you need nematode tolerance. The wild rice, some of the wild rice, not all, are immune, where the cultivated has got huge infestation. Now I'll give you only one example. There are several examples, and the same story can be repeated for any gene which you want. The trait is there, gene can be found out, and it can be transferred in already adapted mega variety. So this is one example here for the salt tolerance in MKSWR 173. We make BC1 में हमने जीनोटाइपिंग किया, BC1 F2 में phenotyping करके ऐसे. Then we identify no time to go into detail here, the 5 QTLs from that one variety, and then it was transferred to cast marker into IR64. Now they have become, I think the salinity tolerance is not an issue, because in the mega variety we have transferred major QTLs for seedling stage as well as for reproductive stage tolerance, and these have been released. Now that was the rice story. Very quickly we go to the pigeon pea story, because pigeon pea is our crop. Nobody will spend their money around the world. So we did, with the support of ICR, Indo-Swiss में इसको we sequenced it. Then now we have finished it to reference quality. Just this year, the ICR का जो reference genome है, इसने ICAR के genome को replace कर दिया है. ICAR ने भी same time publish किया था. It was as incomplete as ours, but it was claimed as reference genome. But now this has replaced at NCBI ICAR sequence. Now this is reference, international reference for the ASA genome. Now very few gaps are there. Very complete those who are doing. We have done extensive review what is needed in pigeon pea, just published in Journal of Experimental Botany. You have to have really aim. This is the diversity map of the global diversity map of the pigeon pea. ICAR has done a lot of work earlier on species collection, germ plant collection, but it has to be utilized actually. And stakeholders, see the varieties, earlier variety very tall one, the new ones are the dwarf one, because now you have to harvest with the machine, you don't have labor. All the what is needed has been documented now, and this is a very good review involving everyone in the stakeholder, including IIPR, and social scientists were involved. Dr. Alka Singh from IRI, who done the field survey. And then we have the pigeon pea IDO type. अगर हम ये ब्रीड करेंगे तो 4 टन पर हेक्टेयर होगा 4 महीने में. अगर हम इसको ड्रीम में रियलिटी में कन्वर्ट कर दें, and the genes are there. We have identified some dwarfing genes like you have wheat and rice. पिजन पी में भी आइडेंटिफाई कर लिया. इसका चिप भी बन गया, QTL भी आइडेंटिफाई हो गए, बट इनको ट्रांसफर करना है. और जब 4 QTL एक साथ करने होंगे तो पॉपुलेशन साइज बड़ा होना चाहिए, प्रोग्राम होना चाहिए. केवल ड्रीम करने से नहीं होगा, एक्चुअली वहां पे करना पड़ेगा. देन दिस इज द प्रोग्रेस थ्रू द इंडो-स्विस प्रोग्राम, जहां से वो 6 वैरायटी निकली है. देन मैंगो में सेम थिंग, मैंगो इज आवर ओन क्रॉप, व्हाई समबडी एल्स विल स्पेंड मनी. तो इसको भी हम लोगों ने जीनोम सीक्वेंस या QTS वगैरह सारे आइडेंटिफाई किए हैं, चिप भी डेवलप किया है. Very high lot value QTS are there. Now they have to be used for the improvement of mango. Mango में you cannot do breeding like rice and wheat. So genome editing is the option there. Because अगर आपको आम्रपाली के स्किन को रेड करना है, तो रेडनेस का जीन लेके आम्रपाली में डाल देंगे, फिर रीजनरेट कर दीजिए, हो जाएगा. अगर आप क्रॉस ब्रीडिंग करेंगे तो 5 साल तो जुवेनाइल हो जाए, और इसके बाद सेग्रीगेशन इतना होगा कि वापस आम्रपाली नहीं मिलेगा. So that's why the new technology, it is essential that we adopt new technology. अगर आपको incremental improvement over the best material करना है, तो वो GM और genome editing से होगा. Cross breeding से भी होगा, तो back cross breeding, but that also will take time. So we have identified several genes for mango also. लेकिन उस gene का होगा क्या? What will you do with that gene unless you have a breeding program? So right from the conservation to evaluation to identification of gene to integration into the highly adapted background, ये पूरा चेन कंप्लीट होना चाहिए. जूट का भी, कि जूट भी हमारी क्रॉप और बांग्लादेश का है, पूरा दुनिया में और कोई जूट नहीं उगाता है. तो इसका भी जीनोम करके इसको भी हम लोगों ने रेफरेंस क्वालिटी पहुंचाया है. एंड वी हैव आइडेंटिफाइड जीन फॉर फाइबर यील्ड, फॉर फाइबर फाइननेस, फॉर नाइट्रोजन यूज एफिशिएंसी. ये सब काम हो गया है. तो last slide says, I just wanted to give you a flavor that rice में starting from the genome sequencing, making use of the germ plant, we have identified the genes, and already जो gene identified थे globally, उनको हमने mega variety में डाल करके we are preparing better than mega variety, and which is getting accepted. Same story can be repeated in pigeon pea, jute, mango, etc. Of course, pigeon pea में अभी कोई mega variety नहीं है, तो हम far behind है. So this is this, to sum it up, that is the challenges. The सबसे बड़ा challenge है to still to conserve and preserve our genetic resources, sir, because they are under great threat. और एक बार चला जाएगा तो 1 मिलियन साल पहले जाके फिर इवोल्यूशन शुरू होगा. तो जितना भी बचा है उसको हमें it should be a holy cow, it should be high priority, nothing doing. आपको बिल्डिंग बनाना है, यूनिवर्सिटी बनाना है तो शहर में जाके बनाओ, सुराहा ताल में मत बनाओ. स्कूल बनाना है तो गंभीर बन के ताल में मत बनाओ. Who will tell to the planner? Scientists have to tell and planner has to accept it. Maybe it is not being told to them कि नहीं यहाँ पर गार्ड से कि यहाँ पर मत करो. Then we go through, we use the modern technology. अपने क्राफ्ट्स के लिए अपना जीनोम इस्टेब्लिश करें, जीन आइडेंटिफाई करें और इम्प्रूवमेंट पूरा प्रोसेस फॉलो करें. देन वी कैन बी सेकंड टू नन. हमारा जर्म प्लांट सेकंड टू नन है, वैरायटी में भी हम सेकंड टू नन है फॉर मोस्ट ऑफ द क्रॉप्स. बट जब मॉडर्न टेक्नोलॉजी हम नहीं अडॉप्ट करेंगे तो वैरायटी भी बाहर से आएगी. जो हमारे पास सारी कैपेबिलिटीज हैं, बट स्टिल वी विल बी डिपेंडेंट ऑन द आउटसाइडर्स इफ वी डोंट टेक द चैलेंज. जब इतना बड़ा हमारा ऑर्गेनाइजेशन से सब कुछ रहते हुए, सो माय अर्ज with the system is that let us get on with the job and be the number one in the world, not the number two. In every field, we have everything to make us number one. So thank you very much for your attention and thank you for the opportunity, Dr.
बहुत बहुत धन्यवाद सर. मुझे विश्वास है कि आपके इस वक्तव्य से भविष्य के अनुसंधान के लिए एक मार्गदर्शक प्रकाश स्तंभ सिद्ध होगा. एक बार पुनः आपका दिल से धन्यवाद सर. अब मैं भारतीय कृषि अनुसंधान परिषद के माननीय महानिदेशक एवं कृषि अनुसंधान एवं शिक्षा विभाग के सचिव आदरणीय डॉक्टर एमएल जाट जी से अनुरोध करती हूं कि वे अपने उद्बोधन द्वारा हम सभी को मार्गदर्शन प्रदान करें.
Good morning. Our mentor, a visionary leader, institution builder, a friend of farmers, chief guest of today's program, Padma Bhushan, Dr. R.S. Paroda ji, our dynamic Secretary ICR, Additional Secretary DARE, Gyanendra ji. We just heard Dr. N.K. Singh ji. What a great work. I think for many people like me, we don't understand the QTLs and all those things. But I think the power of the work, what he have presented and done together with the team is marvelous. So my congratulations to you, Dr. Sahab. Our DDG of Crop Sciences and unwilling director of NBPGR, Dr. Yadav, Dr. Gautam, many senior NBPGRians, colleagues and friends. First of all, let me congratulate you all on this 51st Foundation Day of this great institution. We celebrated 50th, so I was here. I'm very lucky. We're celebrating the whole Diamond Jubilee of the institution. When I think about this institute, and when I think about the people, how they perceive about the institution, and when I think about what they do and what they have contributed, what is going on, it's totally a different story. For an outsider, NBPGR means they are collecting something and storing something. That's what people generally know. But I think the NBPGR is the foundation for our future. Now, two aspects. One on the science excellence, two on preparing ourselves for the future. That means cultivating the leadership. And if you look at the contribution of this institute on both aspects, it's unparalleled. We talk about environmental footprints, but we generally don't talk about the institutional footprints. And I think somebody need to put the footprints on a global map. What the leadership of this institute have done across the globe. Where are the footprints? Cultivating leadership is a greatest challenge today. But look at this institute, which really cultivated leaders, not only the science. And I think that is the significant contribution of this institute. You know why this is happening? It's because this institute work like a family, which is reflected even today. You go to any of those institutions where the foundation day is celebrated, when many of the past employees or the directors are not there. But here, what I see, that they are attached by heart. And I think that makes a difference. And everyone who worked here, who nurtured, who built this institute, have done by heart. That's why this institute is unique. I think the 50 glorious years, and we can look back what we have done. But I think we are a bit shy in documenting the larger impact. If somebody can really do a systematic study, what is the impact of the NBPGR, just not collection and conservation of the germplasm, but where that germplasm fed to and how that translated, what Dr. N.K. Singh mentioned about a lot of those stresses. If someone is making use of that germplasm or the gene which is available in that germplasm. So I think somebody need to put effort to document that. And I tell you, it is unparalleled. Because whatever we claim is coming from somewhere. We generally talk about the unsung heroes. And here are the unsung heroes. There's a lot of background work. And somebody released the variety, and those unsung heroes are not counted. But there is a significant role. If you don't go to those sites and collect the wild relatives from the school side, from the road side, where we are almost missing those, you can't have that gene and then use it in our crop improvement programs. The institute got global recognition, not only because of the germplasm exchange, but this institute and its leaders have built many of those gene banks, and our hero is sitting here, Dr. Proda, who I don't remember how many gene banks he built in Central Asia and other places and guided and helped rest of the world. And that's why even the ICAR Gene Bank named as Dr. R.S. Paroda Gene Bank. I don't know what is the name of this gene bank, but ICAR Gene Bank is Dr. R.S. Paroda Gene Bank. I think the future is not Koji. And if you talk about a secured future, it's in your hand. If you look at the challenges, the climate or the biodiversity was mentioned, all the other aspects. And from that perspective, this germplasm, not only the conservation, but its use, is going to play a very, very important role. That's why I say future is in your hands. We're talking about breeding for tomorrow. And breeding for tomorrow essentially means how best we can make use of the germplasm available in our gene bank or whatever we can get from wherever. But then use is something important, and that's where this institute, I feel like, is a foundation for all the crop improvement program. But NBPGR in isolation and the crop improvement institutions in isolation is not going to help us. So we truly need to have a strong connect to make use of that germplasm, pre-breeding. Now that connect is pre-breeding. From gene bank to the crop improvement institute and in between is pre-breeding. We have to strengthen the pre-breeding. That's where the role of NBPGR and the crop improvement institutions comes into the picture. That means joining hands together and working on that aspect. We truly need to strengthen the pre-breeding. If we don't do that, then we will not have many encasings in the future. I think this is something very, very important. I think I indicated last time also. We talk about the discovery. But I think discovery to delivery. So when we talk about the trait discovery and we talk about delivery, that means seed system. So the trait discovery to the seed system, it should be connected. That connection is something important if we really want to make an impact at the ground. And that's where the connectedness is super important. Now, trait discovery, we got all those tools. I'm not going to, talk, you all know about. But then those tools should not be used in isolation. Person sitting somewhere using a tool but don't have idea about the germplasm. So I think it's something important. But at the same time, we also need to think about the data available with the gene bank, all the passport data, how the breeders can make use of that data, and that's how we can make use of those analytical tools, AI or whatever you call. But I think for that, we need to have a system in place, maybe a dashboard, where breeders can see, assess, or somebody can help them to assess and then pick a specific aspect for a specific trait or a specific a crop or whatever, specific geography. And that's where we can't afford to do all the conventional way what we've been doing, because we did that, but time was different. Today it's evolving and changing very, very rapidly. And we have to keep pace with that evolution process, that time. And if we don't catch the time, we will be losing. And we don't want to lose. And that's where a good mix of the evolution of the science, the new tools and techniques, and our traditional wisdom and the work, I think that's where the connection is something very, very important. And from that perspective, NBPGR is not a stand-alone institute. It's a foundation, enablers, a cross-cutting for all our breeding and crop improvement programs. And I think if we can think around that, will be really helpful. Few days back, under the chairmanship of Dr. Parodaji, we had this meeting of the board of National Advisory Board of the Plant Genetic Resources, including all our bureaus. And I think bringing all those things together is also something important. That's another dimension, not only the plant genetic resources, but other resources. And plant genetic resources got a connection with whatever genetic resources you talk about, whether you talk about fish or animal or microbes or whatever. And that connection is something important. And I think we need to think, can we make a program connecting all those aspects? What is the interconnectedness of different genetic resources? And there is, because we talk about One Health. Now, in the context of One Health, if you just look at the plant genetic resource, I think we will not be able to do that One Health justice. That's where we need all the connection. But then the interconnectivity is something important, not working in isolation, making your own, pieces and try to bring them together and assemble. You can't assemble those because they were developed in isolation. There is no way they can connect. So we need to think about that. I was so happy when Dr. N.K. Singh said about PGNP. I think the oilseed pulses, cotton, millets, those are our priority crops. And that's where we really need to dig out not only the germplasm, but the genes in the germplasm or the QTLs or whatever you talk about, how to break the barriers, the breakthrough in terms of these crops, which is something very, very important. So we truly need to focus on those aspects. And from that perspective, I'm requesting, people like Professor N.K. Singh to mentor some of the people in terms of the gene discovery, because that is something very, very important. If you can have few young scientists who can be mentored and they can carry forward the legacy, is something crucial. Community seed banks, I think it's very, very important, specifically for the opportunity crops. And when talking about agriculture for health, the Sehat program, again, all the opportunity crops are going to play a very, very important role. Even if you look at what the targets we put for the Viksit Bharat 2047, some of those targets are already there, that we have to identify and mainstream those opportunity crops into our agricultural system or agri-food system, and help farmers in terms of improving the productivity and income and their livelihood on the one side, but also how that can contribute to the health of the people. So that's another important aspect. Now, a mention was made about staffing. There are some deficiencies. I think the scientist is fine and a good number, but the support staff, so we will work on it and we'll do our best to provide needed support. But we also need to think about how can we do business better? How can we improve the efficiency? So that's another task, and it's not the efficiency. And someone from the headquarters will tell you, do this. I mean, we all are equally qualified. So I think we have to think about, we have to own the whole system and improve the efficiency and make use of. I mean, this institute got a lot of opportunity. For example, IRI is here and IPB is here, all those institutions. So how can we harmonize some of those things and improve the efficiency? But I think DNA, fingerprinting, there was an NRC, but then we put it under a division. But I think we are focusing and refocusing again. So we have to build that system, which is very, very important. I will not be taking a lot of your time, but I think we have a bright future when we look at the germplasm. And our leaders have been helping us, and that's why we are coming up with this safety gene bank, which is going to be our pride. And we all have to work hard to build that, not only build that, but then think from now. We can't wait until that is built, what we will get out of it. What is the trajectory? What are the directions to make good use of that facility? So I think that's another task to all of us on this very, very important day. Let me again congratulate each one of you for all the great work and also cherishing this 50 years of the glorious journey. And let me also sincerely thank Dr. Yadav for nurturing the institution and taking a lot of his personal time for helping the institution. Thank you so much.
बहुत बहुत धन्यवाद सर। मैं चाहूंगी एक बार जोरदार तालियों से सर का स्वागत करें। सर, आपके प्रेरणादायी विचारों एवं मार्गदर्शन के लिए हार्दिक धन्यवाद। आपके दूरदर्शी सुझाव हमारे लिए मार्गदर्शक सिद्ध होंगे। हमें पूर्ण विश्वास है कि आपके मार्गदर्शन का अनुसरण करते हुए आईसीआर एनबीपीजीआर आने वाले वर्षों में नई ऊंचाइयों को प्राप्त करेगा। आज के इस विशेष अवसर पर एनबीपीजीआर द्वारा प्रकाशित कुछ महत्वपूर्ण प्रकाशनों का लोकार्पण भी किया जा रहा है। मैं तकनीकी दल से अनुरोध करती हूं कि कृपया संबंधित विजुअल स्क्रीन पर प्रदर्शित करें। इसके उपरांत मैं माननीय मुख्य अतिथि एवं मंचासीन अतिथियों से इन प्रकाशनों का औपचारिक विमोचन करने का विनम्र अनुरोध करती हूं। We first will release the books from Division of Germplasm Evaluation. These are 4 series: standard operating procedures and descriptors for characterization and evaluation of field crops. मैं रिक्वेस्ट करूंगी सभी ऑथर्स ऑफ वॉल्यूम 1, 2, 3 and 4 से कि वो स्टेज पे आके बुक रिलीज करें। वॉल्यूम 1, सीरियल एंड स्यूडो सीरियल्स बाय डॉक्टर ज्योति कुमारी, डॉक्टर आर.के. गौतम, डॉक्टर मोहर सिंह, डॉक्टर विकेंद्र कौर, डॉक्टर राकेश भारद्वाज, डॉक्टर संदीप कुमार, डॉक्टर अंजू महेंद्र सिंह, डॉक्टर जीपी सिंह एंड डॉक्टर डी.के. यादव। वॉल्यूम 2, लेग्यूम्स, डॉक्टर कुलदीप त्रिपाठी, डॉक्टर गयाचरण, डॉक्टर राकेश भारद्वाज, डॉक्टर पद्मावती गोरे, डॉक्टर आर.के. गौतम, डॉक्टर अंजू महेंद्र सिंह, डॉक्टर जीपी सिंह एंड डॉक्टर डी.के. यादव। वॉल्यूम 3, ऑयल सीड बाय डॉक्टर लक्ष्मी यादव, डॉक्टर आर.के. गौतम, डॉक्टर सुनील गोमाशे, डॉक्टर विकेंद्र कौर, डॉक्टर ममता सिंह, डॉक्टर सपना लांगियान, डॉक्टर जे अरविंद, डॉक्टर अंजू महेंद्र सिंह, Dr. G.P. Singh and Dr. D.K. Yadav, and the last volume on millets by Dr. Badal Singh, Dr. Jyoti Kumari, Dr. Mamta Singh, Dr. Sunil Gomashe, Dr. Venkatesh K, Dr. Sapna, Dr. Bharadwaj, Dr. R.K. Gautam, Dr. Sushil Pandey, Dr. Anju Mahindra Singh, Dr. G.P. Singh and Dr. D.K. Yadav. A big round of applause for them. One book was released from ICAR-NBPGR Regional Station, Ranchi, Jharkhand, on a living repository of ethnobotanically important plant wealth at ICAR-NBPGR Regional Station, Ranchi, Jharkhand. The authors are Dr. N.S. Pawar, Dr. K.C. Bhatt, Dr. Monu Kumar, Dr. Rakesh Bharadwaj, Dr. Amit Kumar, Mrs. Neha Sharma, Dr. Sadhna Maurya, and Dr. R.K. Gautam. I request all the authors to kindly come on stage. Please be fast. हमारी नेक्स्ट बुक और ब्रोशर है कम्युनिटी सीड बैंक पे ऑपरेशन एंड मैनेजमेंट ऑफ कम्युनिटी सीड बैंक बाय डॉक्टर पीके सिंह, डॉक्टर एसके मलिक, डॉक्टर स्वयं चितन, Dr. Subarna Hajong and Dr. Krishna Kumar. I request all the authors to kindly step on the stage, please. ये बुक हिंदी, अंग्रेजी और असमीज में रिलीज हुई है. डॉक्टर एसके मलिक प्लीज. इसके पश्चात जर्म प्लाज्म एक्सप्लोरेशन डिवीजन से 3 किताबें रिलीज हो रही हैं. पहली है लोकार्ट, स्टेटस ऑफ जेनेटिक रिसोर्सेज एंड कल्टिवेटेड वेराइटीज इन इंडिया बाय डॉक्टर पवन कुमार मालव, डॉक्टर प्रवीण कुमार सिंह, डॉक्टर पंकज कुमार कनौजिया, डॉक्टर ओपी धारीवाल, डॉक्टर अनिल पटीदार, डॉक्टर नवप्रेम सिंह, डॉक्टर एस के मलिक एंड डॉक्टर डी के यादव. The second book is on pineapple. I request all the authors to kindly come on stage. Dr. Julius Uchoi, Dr. P.K. Singh, Dr. Swim Chitan, Dr. P.K. Kanojia, Dr. S.R. Padi, Dr. G.D. Harish, Dr. D.K. Yadava. Third book is on Chironji by Dr. D.P. Sembal, Dr. K.C. Bhatt, Dr. O.P. Dhariwal, Dr. P.K. Singh and Dr. D.K. Yadava. I request all of them to kindly come on stage. A huge round of applause for them. The next book in the line is the edited one from the Division of Plant Quarantine. Potential quarantine pest for India in root, bulb and tuberous vegetable crops. Editors are Bharat H. Gavde, Dr. Sangeeta Pandey, Dr. Pooja Kumari, Dr. Kavita Gupta and Dr. V. Celia Chalam. I request all the authors to kindly come on stage quickly. जोरदार तालियों से इनका स्वागत किया जाए. हमारी आज की बुक है, लास्ट बुक जो रिलीज हो रही है वो है डिवीजन ऑफ जर्म प्लाज्म कंजर्वेशन से इन्वेंटरी ऑफ रजिस्टर्ड क्रॉप जर्म प्लाज्म 2021 to 2026 बाय डॉक्टर अंजू महेंद्र सिंह, डॉक्टर अंजलि काकौल, मिस्टर अरूप दास एंड डॉक्टर डी के यादवा. बहुत बहुत धन्यवाद सर. कृपया अपना स्थान ग्रहण करें. अब मैं एनबीपीजीआर के माननीय निदेशक डॉक्टर डी के यादव सर से विनम्र अनुरोध करती हूं कि वे हमारे माननीय मुख्य अतिथि पद्म भूषण डॉक्टर आर.एस. परोदा सर को शॉल एवं स्मृति चिन्ह प्रदान कर सम्मानित करें. धन्यवाद सर. मैं पुनः आपसे आग्रह करूंगी कि हमारे विशिष्ट अतिथि आदरणीय डॉक्टर एमएल जाट जी को शॉल एवं स्मृति चिन्ह भेंट कर सम्मानित करें। सर से पुनः अनुरोध है कि वो हमारे विशिष्ट अतिथि श्री ज्ञानेन्द्र देव त्रिपाठी जी को शॉल एवं स्मृति चिन्ह भेंट करें। मैं पुनः डॉक्टर डी के यादव जी से अनुरोध करती हूँ कि वे हमारे आमंत्रित वक्ता आदरणीय डॉक्टर नागेंद्र कुमार सिंह जी को शॉल एवं स्मृति चिन्ह भेंट कर उन्हें सम्मानित करने की कृपा करें। सभी सम्मानित अतिथियों का हम हृदय से अभिनंदन एवं आभार व्यक्त करते हैं। अब इस समारोह के सर्वाधिक महत्वपूर्ण क्षणों में से एक की ओर बढ़ते हुए मैं हमारे माननीय मुख्य अतिथि पद्म भूषण डॉक्टर आर.एस. पड़ोदा सर से सादर अनुरोध करती हूँ कि वे अपना उद्बोधन प्रस्तुत कर हमें अपने बहुमूल्य अनुभवों, विचारों एवं मार्गदर्शन से अनुग्रहित करें।
मेरी तरफ से आज गोल्डन जुबली के समापन समारोह पे आप सभी को बहुत बहुत बधाई। एनबीपीजीआर में आके ऐसा लगता है कि फैमिली में सबसे फिर वापस आके जुड़ गए हैं, बहुत खुशी है। और खुशी इस बात की है कि मंच पर जो है नए एनर्जेटिक डायनेमिक डायरेक्टर जनरल डॉक्टर एमएल जाट हैं, डॉक्टर एनके सिंह हैं जो माने हुए वैज्ञानिक हैं जिन्होंने बहुत कुछ कार्य अपनी लाइफ में किया है और इसके लिए हम इनको बधाई देते हैं। डॉक्टर डी के यादव, आपने भी यहाँ बहुत कुछ योगदान दिया है और अब वो मुझे कहा गया था कि अनविलिंग डायरेक्टर हैं, तो ज़रा विलिंग हो जाइए जब तक रेस्पॉन्सिबिलिटी है। और डॉक्टर आर के गौतम, हमारे जो है विशिष्ट ऑल्सो अतिथि श्री ज्ञानेन्द्र त्रिपाठी जी जो आईसीआर के सेक्रेटरी हैं और उन्होंने भी अपने उद्बोधन में काफ़ी अच्छी बातें बताई। साथ ही यहाँ उपस्थित कई मेरे पुराने साथी हैं, हो सकता है मैं नाम लेने में चूक जाऊं, पर डॉक्टर करिहालू हैं, डॉक्टर उमेश श्रीवास्तव हैं, डॉक्टर अशोक कुमार हैं, डॉक्टर प्रशांत दास हैं, डॉक्टर भागमल जी हैं, डॉक्टर पी आर चौधरी हैं, विश्वनाथन जी चले गए हैं, डॉक्टर अनुराधा अग्रवाल, डॉक्टर वीना गुप्ता जी हैं, डॉक्टर रोशनी नायर हैं, डॉक्टर जेसी राणा हैं, डॉक्टर एस के मलिक हैं, बीएम सिंह हैं, सुधीर कोचर हैं, डॉक्टर अहलावत हैं, डॉक्टर एस के मलिक हैं, डॉक्टर सुनील अर्चक जो प्रोफेसर हैं इस इंस्टीट्यूट में जो प्लांट जेनेटिक रिसोर्स को देखते हैं पढ़ाने के लिए। So let me say I am very privileged to be here today and to participate in this concluding celebration of the Golden Jubilee of this great institution that we have built over the last 50 years. 50 years in the life of an institution is not merely an anniversary. It is an opportunity to celebrate and to have an introspection for its renewal and reinvention. During these 5 decades, NBPGR has quietly built one of the India's greatest strategic assets, the nation's genetic wealth. Long before climate change, nutritional security, and biodiversity conservation related threats emerged, NBPGR did recognize that the future of agriculture begins with conserving its past heritage. Today, India possesses one of the world's largest collections of plant genetic resources. I congratulate you all who are working day and night to see that it is conserved well and scientifically. Behind every accession lies the wisdom of generations of farmers, explorers, breeders, and scientists. This extraordinary legacy belongs not only to Indian agriculture, but to the entire humanity. I congratulate every scientist, staff member, and former directors, various stalwarts who supported it, like Dr. B.P. Paul, Dr. M.S. Swaminathan, Dr. A.B. Joshi, Dr. Harbhajan Singh, Dr. Hardas, Dr. Mehra, Dr. R.K. Arora, and many of those colleagues who are sitting here. Yet, while we celebrate 50 years of our achievements, we must also ask now a more important question. Will the next 50 years look anything like the last 50? Or the business will remain as usual? I believe the answer is no. The world of plant genetic resources is currently entering its most transformative period ever. The 4 transformational forces we need to address, therefore, today are climate change, digital agriculture, molecular biology and genome editing, and institutional transformation. So, when we think of climate change is changing not merely temperatures, but it is changing the genetic requirements of agriculture itself. Tomorrow's varieties cannot be bred from yesterday's diversity alone. We shall need new sources of tolerance to drought, heat, flood, salinity, and emerging pests. Those genes are increasingly found in crop wild relatives, traditional land races, and underutilized crops. Dr. N.K. Singh's presentation clearly support this without any doubt. And therefore, I am reminded of the most famous article of Dr. B.P. Paul, which was having its title, Search for New Genes. So search for new genes will continue, and we would really need much more important genetic diversity, which will address the emerging challenges. Fortunately, India is bestowed with one of the most unique genetic resources, be those plants, be those animal, be those fish, be those forestry trees, be those grasses, and be those horticultural crops. The best tolerant genes for salt, like Pokali rice and Kharchia wheat, come from India, and we have given to the world. Even for submergence, the sub1 gene comes from Indian germplasm. And today, Swarna sub1 is one of the best examples. And now IRI is taking benefit of it to support others in the world. So every accession conserved today is potentially tomorrow's insurance policy against variable kinds of climate uncertainty, be those floods, drought, frost, or terminal heat. Second part, strengthen our digital sequence information. We are entering an era where genetic resources are no longer only physical. Increasingly, they exist as digital information. It is very clear now that whosoever governs digital sequence information will increasingly govern agricultural innovation and ownership. What are we doing in this area and how much more is required to be done? I think efforts have been made. Sunil Archak possibly can vouch for it. And we had deliberations at national level, and there is definite need to move forward. The third aspect which I mentioned was molecular biology and genome editing. Genome editing, artificial intelligence, and synthetic biology are not threats to agrobiodiversity. When properly governed, they become powerful tools for unlocking the hidden treasure of our genetic wealth. Conservation and innovation must therefore move together, not in opposition, but in partnership. No doubt, for harnessing and scaling innovation, we shall need much stronger public-private partnership with clear understanding for access and benefit sharing. There is opportunity for generating resources through this kind of partnership, and institutions like NBPGR can provide the great leadership. Next is the institutional transformation. We should not just be happy on the past laurels. We are proud that we have the world's second largest and most modern gene bank. But when was it built? Almost 30 years ago. And any building's life is treated as 40 years. So if you don't again remodernize it and take care, possibly we will be missing an opportunity. The gene bank has recently been renovated. I am happy because we worked for it for almost a decade, and glad that ICAR allocated funds. And those funds left for almost 4 or 5 years before the gene bank could be renovated. Imagine if engine stops working for the gene bank, which requires minus 18 degrees Celsius temperature, then what will happen to our national heritage? So we have to make sure that it functions, and I'm proud that it has never failed. So for too long, conservation and utilization have proceeded on parallel tracks. We can't continue working on parallel tracks. Dr. M.L. Jat often talks about no time now to work in silos. So we have to make sure that within institute itself and in ICR, the conservation through use becomes our great focus in future. The true success of a national gene bank is not measured by the number of accessions stored, like black box, but by the number of farmers whose livelihood improve because of those genetic resources were quite effectively utilized. Every conserved accession should eventually contribute to evolve better varieties, possessing better nutrition, greater resilience, and higher income for the farmers. You would agree with me that the conservation without utilization is merely preservation. Hence, the conservation with innovation leads to national development. Now, I would come to another aspect that we need to not only become but remain a global leader in the area of plant genetic resource management. India is uniquely positioned to become a global leader in plant genetic resources. We are one of the world's richest centers of crop diversity. We were told we have more than 4.7 lakh accessions in our gene bank. We are happy that we built the gene bank with a capacity of 1 million 30 years ago, and in 30 years we could only fill half of it. So at least we can safely go on working and conserving more till 1 lakh number is achieved. But then what after that? We also have a strong network of scientific institutions, unparalleled elsewhere. 6 bureaux within the ICR. You won't find in any developing or even I would say developed country. So imagine institutions, and Dr. Borlaug told me that cradle of success for green evolution had been the institutions and human resource. So we have those cradles now with us. And policy support. So if policy support was not there, these bureaux could have not been created. Since they are now created, we need to nurture them to the best of our ability. We have one of the world's largest agriculture research systems also, with well-trained scientists engaged in plant breeding and biotechnology. And we can take pride, possibly at NBPGR only, when we built this gene bank and this bureau. Not only we had the facilities created, including the equipments, the best equipments, but possibly all 100 scientists and important technicians were trained in the United States. You won't find that kind of example. And unless you go and see you would not understand where you stand. And I think that was a turning point. We also have the most hardworking and visionary farmers. So when Dr. Borlaug said among cadres, I mentioned two. The other two were policy support, and the fourth one was the hardworking, willing farmers to adopt technology once convinced about the benefit of it. We also have the strong political support, as I said, to conserve and use our genetic resources for food and nutritional security as well as for posterity. This gene bank was inaugurated by Dr. K.R. Narayanan at that time, who was vice president and later became president. And when he was putting the badge on me while giving Padma Bhushan, he remembered and asked me, how is our gene bank? And please take care of it. We also had Dr. APJ Abdul Kalam visiting this institute. And in this hall, he pronounced that whatever I have been working had been for the destruction of humanity. But after coming here, I realized that what you are working is for posterity. And if there is any rebirth, I would like to be an agricultural scientist. So now also, when our Prime Minister inaugurated the first Agrobiodiversity Congress that we happened to organize, all of you, he had also released the Delhi Declaration and pronounced that all support will be given for realizing it, which has many specific recommendations, and I hope you all are looking at it and taking care of, especially the use of wild and weedy materials and germplasm indexing and partnership at regional and global level. Recent declaration by Prime Minister, not by anyone else, directly by Prime Minister, of rupees 500 crores for building the duplicate safety gene bank, announced almost, I would say, year and a half ago. Again, reflects how much support we have for the kind of work that these colleagues sitting in this hall are working. And PM is clear, the announcement by PM is a clear indication that India's biological heritage is a strategic national asset. However, the second national gene bank should not simply replicate the first, as someone were talking when money was given, that we should have it in Hyderabad. It actually must become a digital, climate smart, and globally networked facility integrating genomics, artificial intelligence, cryobiology, phenomics, and real-time based information system. Comparable to or even better than Svalbard Global Seed Vault that was built in Norway when we established Global Conservation Diversity Trust, with which I was associated for generating resources and we could at that time get about $120 million. We would like to see that this kind of vault is nowhere else in any developing country than in India. So that should be our goal. And therefore, I would urge to Honorable Director General, Deputy Director General, to please give high priority and not let a day be wasted for constructing and creating it, so that when we have centenary celebration of ICR in 2029, this is dedicated to the nation. These strengths together make India not merely a custodian of valuable biodiversity, but the world's leading innovator in agrobiodiversity-based agriculture. So, I would like to now come to my final part, that we must now think of our future roadmap. And for that, there are certain important recommendations. And let me now suggest Saptarishi, 7 sage today. And these are complete collection of all crop wild relatives. First. Second, modernize once again the National Gene Bank. Third, build India's digital sequence information platform so that we don't lag behind others. Fourth, strengthen DNA fingerprinting and cryopreservation initiative. We started with it, Dr. Karihalu. I think that is your worst disappointment, that National Research Center on DNA fingerprinting that was created that time got closed. I hope we revive it, and under PVP FRA Act for the dust test, we are now proposing, because that act also now is being revised, and our committee has given recommendations where we have clearly stated that DUS test must be based on DNA fingerprinting and molecular biology. So that I think we need to take care. Fifth, promote community seed banks was talked about, and on-farm conservation, the kind of work which Biodiversity International and PVP FRA is doing requires to be also brought in the mandate of NBPGR. It is not ex situ collection alone. Lot of the genetic variability is being protected by our gene saviors and tribals. They need support, and they need support for not only the facilities, but also for human resource and also for their livelihood through value addition of those genetic resources and marketing those for larger benefit to the community. The recent announcement by the Gene Bank under PVP FRA, I am happy that that has happened. We had been wanting it for long, and we want that at least that gene fund should be minimum of 100 crores. Fortunately, now it is about 20 crores. Dr. Mahapatra could not join today because of his other personal problems, but the announcement has been made to seek proposals for supporting these gene saviors for community gene bank, and training of those for human resource development. This has to be scaled. Scaled because just 20 crores will not be able to do much. Maybe I would urge Dr. M.L. Jat that you have been now focusing on generating resources, emphasizing also of partnership with private sector, and taking benefit of CSR, the corporate social responsibility. Opportunities are there, and also your own corpus fund in the ICR. At least 100 crores is earmarked separately for in situ conservation. The sixth, launch a national PGR evaluation mission. I think it is being done to some extent, but the speed is slow. Partnership is very minimal. It has to be very aggressive, using networking with all those institutions who have that leadership and have the equipment and facilities like Dr. N.K. Singh. And people like N.K. Singh is a rare resource. Why can't they be professor adjunct or NBPGR under your, postgraduate program? And why can't he continue training people in the area which is so critical and important? The last one, position India as a global leader in international PGR governance. We should not be lagging behind. We have been participating right from beginning. Even I was there when farmers' right under the International Treaty for Genetic Resources for Food and Agriculture was adopted. The farmers' right was recognized for the first time. And then we are happy that in our Plant Breeders Protection Act, we also included Farmers' Right Act. It is a unique one, nowhere in the world. And that time we got that particular act approved by the parliament in the shortest possible time of 5 years. So that kind of leadership at international level. Question is, our Jarplan through CG centers has gone into the Svalbard Global Seed Vault. But is there any legal system which has been put in place so that we can ask them as to whom to share and whom not to share? And what authority CG centers had to partner with that germ plant without seeking approval of the ICAR? I would say it was a kind of bypassing, and by the donors, by the developed nations, to take everything and protect it. Now we are going to build our own Svalbard in Himachal Pradesh. We must see that we provide leadership for the South. So South-South partnership is an opportunity. And regional gene bank has been announced many times, even through SAARC, that India is willing to provide support to all SAARC countries. But we are in a more sensitive political environment. which has not made it possible. But where we have MOUs developed, the countries like Central Asia, where they don't have the human resource and institution, where I tried to build the gene bank in each country, though that was not my mandate. And they are wanting to develop MOU with us, and they have the best genetic resources available with them. And they want to keep their material in our gene bank. But I think we have not been able to take benefit of that kind of opportunity. So as we celebrate 50 years of NBPGR, let us remember that seeds are much more than biological material. They are repositories of evolution, culture, resilience, and our future hope. So coming to my last part, every accession conserved today enlarges India's option for tomorrow. Remember, the future of Indian agriculture will not be secured merely by conserving agrobiodiversity, but by converting it into climate resilience, food and nutrition security, farmer prosperity, and leadership at the global level. This must be our mission for the next 50 years. I have no doubt that under ICR, this institution, which is known as NBPGR, with its outstanding scientific leadership and dedicated team, will continue to stand at the forefront of this national endeavor. Today, therefore, while concluding, we must resolve that when future generations celebrate the centenary foundation day of NBPGR, that is 50 years after, they will say that the scientists gathered here today in 2026 had the right vision to transform India's priceless genetic wealth into one of the greatest engines of agricultural growth and national development. Finally, we all know that the future of Indian agriculture is hidden inside the seeds that we conserve today. Our responsibility is to unlock that treasure for posterity of every farmer, for prosperity of every farmer, every citizen, and every generation yet to come. Thank you and Jai Hind.
बहुत बहुत धन्यवाद सर। एक बार जोरदार तालियों से सर का अभिनंदन। सर, आपके द्वारा दिया गया सप्तऋषि कॉन्सेप्ट आज के भारत को, कल के भारत के सामने जो चुनौतियां आने वाली हैं उससे लड़ने का बहुत स्ट्रॉन्ग जो है कि एक आधार देगा। इस पे हम सभी समीक्षा करेंगे और हम सभी को इसके बारे में आगे बढ़ के सोचने की जरूरत है। सर, मैं आपका धन्यवाद करती हूँ। आपके बहुमूल्य विचारों और अनुभवों ने न केवल हमें संस्थान की उपलब्धियों पर गर्व करने का अवसर दिया, बल्कि भविष्य की चुनौतियों और अवसरों के प्रति हमारी सोच को भी नई दिशा प्रदान की है। हमें विश्वास है कि आपके मार्गदर्शन और प्रेरणा से ICAR-NBPGR आने वाले वर्षों में और भी उत्कृष्ट उपलब्धियां प्राप्त करेगा। एक बार पुनः सर, हम आपका हृदय से आभार व्यक्त करते हैं। इन्हीं शब्दों के साथ आज के इस गरिमामय एवं सफल आयोजन के समापन की ओर बढ़ते हुए मैं जर्मप्लाज्म मूल्यांकन प्रभाग के विभागाध्यक्ष डॉक्टर R.K. गौतम जी से विनम्र अनुरोध करती हूं कि वे अपने औपचारिक धन्यवाद ज्ञापन को प्रस्तुत करने की कृपा करें।
धन्यवाद डॉक्टर वर्तिका। नमस्कार, गुड आफ्टरनून। I'm happy to share that we are little ahead of time. So I can take little liberty of time. Yes, sir. Yes, yes, sir. Very fast, sir. Yes, sir. Yes. So it is my profound privilege to propose the vote of thanks on the auspicious occasion of the Golden Jubilee and 51st Foundation Day of ICAR National Bureau of Plant Genetic Resources. On behalf of entire ICAR NBPGR family, I express our heartfelt gratitude to our Honorable Chief Guest, Padam Bhushan Dr. R.S. Parodaji, President TAS and former Secretary DARE and DG ICAR for gracing this historic occasion and inspiring us with his visionary address. Sir, your immense on behalf of entire ICR and DPR family, I express our heartfelt gratitude to our Honorable Chief Guest Padma Bhushan Dr. S. Parodaji, President TAS and former Secretary DARE and DG ICR for gracing this historic occasion and inspiring us with his visionary address. Sir, your immense contribution to Indian agriculture and your enduring association with ICAR and BPGR continue to inspire generations of agriculture scientists. I sincerely express our gratitude to our Honorable Chairman, Dr. Mangilal Jarji, Secretary DARE and DG ICR for his motivating address and unwavering support to agriculture research and innovation. I also extend my sincere thanks to Shri Gyanendra Dev Tripathi ji, Additional Secretary DARE and Secretary ICR for his gracious presence, continued guidance, and encouragement to the ICR system. Our heartfelt appreciation and gratitude to Professor Nagendra Kumar Singh ji for delivering the 13th Dr. Harbhajan Singh Memorial Lecture and sharing valuable insights on the role of genomics in the context of plant genetic resources. I also express our sincere gratitude to Dr. Devendra Kumar Yadav ji, Deputy Director General Crop Science, ICR and Director ICR NBPGR for his visionary leadership, continued support and for presenting the Bureau's achievements during the last 1 year. I extend our sincere thanks to all the distinguished dignitaries, ADGs, former directors, especially Dr. S.K. Sharma ji has joined online, directors and representatives of ICAR Institute, state agriculture universities, national and international organizations, invited guests, and members of the press and media for making this celebration memorable. My heartfelt appreciation and gratitude to all the former staff members, all heads of divisions, OICs, the scientists, administrative staff, administrative staff, technical supporting staff, and research scholars, students, and all contractual staff of ICAR-NBPGR, including regional stations, for their presence and support. Last but not the least, my sincere thanks to all the chairpersons and every member of the organizing committee whose dedication and teamwork ensured the grand success of today's event. As we celebrate the glorious journey of 50 years of ICAR-NBPGR, we seek the best wishes, blessings, and support of all the dignitaries for taking ICAR-NBPGR to new heights for its dedicated service to the nation. Let us reaffirm our commitment for conserving and utilizing plant genetic resources for ensuring food, nutritional, and environmental security for the present and future generation. With these words, I once again thank each of you for your valuable presence, contribution, and continued support. But before I leave, I want to make 3 announcements. One is, we'll have a group photo in the center lawn, and there will be refreshment in the DPQ lawn for all the participants. I also request all the esteemed former staff members to please collect the token of our affection and memory from our team in the center lawn. So with this, I thank once again all of you. Thank you very much. Jai Hind.